Hello students, welcome to Vedantu NEET English. This is Basava Sir, your biology master teacher students. Hello, welcome to your own channel students. Students, quickly let me know in the chat right now if I am audible and visible to each and every one of you. Quickly let me know if I am audible and visible to all of you students. Quickly. Sound check students, sound check. Yes, quickly. Sound check. Quickly let me know students if I am audible and visible to all of you. Quickly. Yes. Now students, this is the day 2. Yeah, day 2 of your entire new series, that is 0 to Hero series, right? Now here we'll be in today's class we'll be detailedly studying the entire your cell, the unit of life, as well as the structural organization chapter. In detail we'll be doing today students, in detail. Okay students, can I get some fire emoji in the chat to let me know that, sir, yes, we can start today's class without any halt. The reason we were late because students, I was stuck in traffic as usual, because I came to office today and it is so much of traffic in Bangalore and you cannot go anywhere. Okay, so hello, good morning students, good morning. Okay, now students, I want everyone to look around you and tell me what do you see. All of you quickly in the chat, I want everyone to look around yourself and tell me what do you see. For example, let's say here I have a glass of water, I have a pen here, and then there are no insects here. There is me, there is camera, there is board, there is light and everything, right? Now students, is all of these things, right, living or non-living? You tell me, sir. Water is non-living, pen is non-living, light is non-living, camera is non-living, and as well as this board also is non-living in nature. Non-living in nature, students. Okay, now let's take me, all of you, the cockroach under your bed, the spider on your wall, the lizard on the top ceiling. Is all of these things living or non-living, students? Quickly in the chat, students. Do you think all of these things are living now? Yes, these things are living. So the first thing which we need to understand here is the difference between living and non-living. So all the living organisms, students, all the living organisms have something in common, right? They have something in common which is not present in your non-living things, students. Who can tell me what is it? Who can tell me what is present only in living organisms? That is nothing but cells. That is nothing but the cells. And these cells, students, these cells are completely absent in the case of non-living things. They are completely absent in your non-living things. Okay. Now, students, is it possible to have an organism made up of a single cell? Students, is it possible? Is it possible to have, like, one organism, it is made up of only one cell? It is possible, students. It is possible. Okay. So, if an organism, if it is made up of a single cell, I will call it unicellular. I will call it as unicellular in nature. Now, what if an organism, let's say humans, if they are made up of many cells? If they are made up of many cells, what do you call them, students? I will call them multicellular. I will call them, students, multicellular in nature. Multicellular in nature. Now, can I get a quick example of, students? Can I get a quick example of unicellular organisms in the chat? All of you. Students, quickly. Hello, Kaushik. Hello, everyone. Can I get an example of your unicellular organism? You will tell me, sir, bacteria. Acute bacteria is an example. Acute bacteria is an example. Now, students, can anyone tell me, is amoeba also an example? You're like, yes, sir, amoeba. So, if you think, students, if you know about amoeba, can anyone tell me which of the following kingdoms do amoeba belong to? Tell me in the chat, students. Tell me in the chat. Amoeba belongs to which kingdom? Quickly in the chat, students. Okay. Now we understood that there is something called as a cell, which is present in all the living organisms. It is absent in the case of your non-living things. It is absent in the case of your non-living things, students. It is absent in the case of non-living things. Clear? Now, students, but what exactly is the cell? What exactly is the definition of a cell? Can anyone tell me? See, if you are in class 10th or something, you will tell me, sir, cell is nothing but, it is nothing but, students, the functional unit of life. It is a functional unit of life. Yes or no? Yes or no? Also, you will tell me, students, it is the structural and functional. We'll add structure also here. We'll add structural also. structural and functional unit of life. But apart from the students, if I tell you a definition, let's see how many of you can agree with me. Right? I will tell students cell is nothing but a smallest structure which is capable of independent existence. All of you agree with me, students? It is a smallest structure. It is a small structure, okay, capable of, it can show independent existence. Independent existence. It is a small structure of independent existence. Independent existence. Okay. Now, for example, students, like, can a cell do something called as, students, respiration? Yes, it can. Can a cell do excretion? Yes, it can. Can it do something called as your transport of gases? Yes, it can. It can do everything. Students, is my voice not clear? Students, is my voice not clear? Or is it clear to all of you? Sound check, students. Sound check. Sound check. Sound check. Yes, it can replicate itself also. It can replicate itself also. That is the beauty of a cell. Now, students, all of you have some idea about a cell right now. Some idea about a cell right now. Okay. Now, can anyone tell me who gave the cell theory? Before I go further, do any one of you, if you have watched my previous lecture, you would know by now, who gave the cell theory, the concept of cell theory? Anyone who can tell me? The concept of cell theory was given by whom, students? Quickly, students. I'm audible, right? I'm audible, right? All of you? Yes, I'm audible. Students, can anyone tell me who gave the concept of cell theory? Anyone, students? Anyone? Who can tell me? The concept of cell theory was given by, initially, by two main scientists. Okay. One was a German, one was a British. The first one is a German botanist called as Matthias Scheiden, and other one is called as a British zoologist. His name is your Theodore Swan. Now, students, you tell me, if he is a botanist, obviously he'll work on plants. Right? That is, he found that in plants there are different kinds of cells. In the case of plants, there are different kinds of cells are present. Now, can anyone name any few tissues in your plant? Any tissues? Anyone? Any tissues in the case of your plants? Sir, you will tell me, sir, in the case of plants, there is parenchyma, collenchyma, sclerenchyma, right? Xylem and phloem. So all of these are different tissues which are found in the case of plants. And this was found by your Matthias Scheiden. He told us that plants are made up of different kinds of cells. Okay. Now, what did your Theodore Swan do? He told us that the entire cell has a covering around it. See, that time we did not know what is the covering. Right now we know that what Matthias Scheiden was referring to was nothing but the cell membrane. So he was referring to nothing but the cell membrane. Cell membrane is a covering around the entire cell there, entire cell. Then, students, what is cell wall? He also told us that the cell wall right here, it is found only in the case of plant, not in the case of animals. So it is an exclusive feature of your plants only. And he also told us that all of us, animals and all the plants, we are made up of cell and product of cells. So we are made up of cells and product of cells. That is all. So this was cell theory given initially by your Matthias Scheiden and Theodore Swan. Okay. But student, this cell theory is okay. In the cell theory, there was no explanation of how cells came into existence. There was no explanation how cells came into existence. Can anyone remind me, students, who was the scientist who came and told us that, wait a minute, you don't know anything, I will explain. Who was the scientist who came and told students, cells actually come into existence from a pre-existing cells? Students, it was none other than Rudolf Virchow. Rudolf Virchow in 1855, he told us that cells divide and new cells are formed from pre-existing pre-existing cells. So from pre-existing cells, we can get new cells. We can get new cells. And he called exactly Jojo. Omnis cellula. He called it, students. What do you call it, students? Omnis, not your omni. Omnis cellula-e-cellula. It means cells exist from pre-existing cells. Right? Pre-existing cells. Omnis cellula-e-cellula. Some Harry Potter spell again, students. Harry Potter spell this is. Okay? Omnis cellula-e-cellula. Clear, students? Clear. Now, so all this is okay, students. But who actually discovered a cell, students? Like, so since we're talking about history, right now, can anyone remind me who was the scientist who discovered this cell? Okay, anyone? Anyone, students? Who discovered the cells in general? Who is this handsome gentleman? Anyone? It was none other than Robert Hooke. Robert Hooke was the scientist who discovered the cells. But. Can anyone tell me in the chat, did he discover living cell or dead cell? Anyone? Robert Hooke discovered the dead cells. All of these are dead cells in nature. Dead cells. Now, if we discover dead cells, can anyone tell me who discovered the living cell then? Living cells were discovered by Anton von Leeuwenhoek. So, Anton von Leeuwenhoek discovered the living cells. Living cells. Now, if a cell is the entire body, imagine a cell is the entire body. There is something called as a brain of a cell. There is something called, students, brain of a cell. Who can tell me what is it? What is the brain of a cell? The brain of a cell is called as the nucleus. And this nucleus was discovered by Robert Brown. So Robert Brown discovered the entire nucleus. Nucleus was discovered by Robert Brown, students. So students, till here, any doubt? Till here, I believe there is no doubt still here, students. I believe there is no doubt till here. Okay. Now, students. Now, this was cell theory. Now, here is an organism called as virus, right? This virus is actually an exception to cell theory. It is an exception to the cell theory. Virus, they're like, I do not follow cell theory. Okay. Now, can anyone tell me, students, this virus right here, is it living or dead? Anyone who can tell me? Who can tell me? Hey, this virus right here, is it living or dead or both? It is actually both, because this virus right here can exhibit living features as well as non-living features. Both can be observed by this stupid or very dangerous virus. Okay, very dangerous virus. Okay, students. Now we learned something called as overview. Now we learned something called as, students, overview of your cell. Now there is something called as a prokaryotic cell as well as a eukaryotic cell. Pro and eu. What is the difference? Understand. Prokaryotic cell is a type of cell which is very primitive in nature or very primitive in nature. Here, the organelles which are present here, right, they do not have any coating around it, or there is no protection. There is no membrane-bound organelles here. So, no membrane-bound organelles in the case of prokaryote. But in the case of eukaryote, students, they have membrane-bound organelles in the case of eukaryote here. Example, nucleus, endoplasmic reticulum, Golgi complex, lysosome. Right? They are membrane-bound organelles. Okay. Now, can I ask all of you one more question? Do you think, students, in the case of your eukaryote, are there any organs which do not have any membrane? Anyone who can tell me? See, I told you, in the case of a eukaryote, we have membrane-bound organelles. But are there any conditions? Are there any conditions in the case of eukaryote where membrane-bound organelle is not present? There are. There are actually two of them. One is called as ribosome. Other one is called as centrosome. So ribosome as well as centrosome, both of these structures do not have any type of membrane around them. Do not have any membrane. Okay, so we can know that ribosome is present in your plants and animals, but centrosome is present only in the case of animals. It is completely absent in the case of plants. Okay, so only animals, centrosome. The main function is it helps in cell division. So all of you know, right, students, how does it help in cell division? All of you know, it helps in formation of spindle fibers. Yes, it helps in formation of spindle fibers, students. Spindle fibers. Okay. No. What is common then? In the case of prokaryote and eukaryote, the common thing is going to be the cell membrane, matrix, that is cytoplasm. It can have genetic material, DNA or RNA, or ribosomes. These are called as the common things in pro and eukaryote. Very simple, students. Very simple. Okay. Now, there are different types of cell size. There are different types of cell sizes here. Okay. First one is called as your mycoplasma, the smallest cell here, that is 0.3 micrometer. Largest is going to be your egg of the ostrich. Longest is nerve cell. Bacteria is around 3 to 5 micrometer. Human RBC is around 7 micrometer here. Okay. Now, students, can I ask all of you a simple question now? Mycoplasma belongs to which kingdom? Quickly in the chat. Mycoplasma belongs to which kingdom? Who can tell me? Kingdom Protista, Kingdom Monera, right? Where do we get your mycoplasma? See, mycoplasma is a Kingdom Monera. It is Kingdom Monera. But unlike your Kingdom Monera, this mycoplasma is very special. Why? Because it does not have any cell wall. The cell wall is completely absent here. Now, one more question to all of you. Mycoplasma, they'll ask you many questions, okay? Do you think mycoplasma, students, has, does it require oxygen to survive? Students, do you think it requires oxygen to survive? It does not. It does not require oxygen. So, in anoxygenous condition, mycoplasma can survive. Okay? So, very important, mycoplasma is very important, students. It's very important. Okay? Now, okay, I'll ask you one more thing now also. Under Kingdom Monera, students, under Kingdom Monera, do you think mycoplasma, do we study it under Eubacteria or Archaebacteria? Tell me that answer in the chat. Do we study it under Eubacteria or Archaebacteria? That also tell me. Okay? No. I can ask you one more question also here, students. Okay? This human RBC, students, human RBC, the size around 7 micrometer. Do you think this human RBC has nucleus? Students, do you think it has nucleus or not? It doesn't have any nucleus. Nucleus is absent in the case of here. Students, do you realize what am I doing? I am trying to connect this chapter to multiple chapters to make you understand this entire concept here. Okay. So, if I tell you, students, if I tell you a statement, Krebs cycle does not take place in the case of human RBC. True or false? Krebs cycle does not take place here. True. Because mitochondria also is absent here. The only respiration, the only type of respiration which takes place in human RBC is glycolysis. That's all. Why, students? Why? Why, students? Why do you think only glycolysis happens? Do you think glycolysis happens in cytoplasm? Do you think glycolysis happens in cytoplasm? Tell me yes or no. Tell me yes or no quickly, students. Okay. Now, there are different types of cells. Red blood cells, white blood cells, columnar epithelial cells, right? Nerve cell, tracheids. Again, students, this tracheids right here, is it a xylem or a phloem? Quickly in the chat, students, xylem or phloem. Then we have mesophyll cells here, mesophyll cells. Okay, mesophyll cells are present here. Students, this mesophyll cells, right? Mesophyll cells can be divided into spongy and palisade. In which tissue? In which monocote dicot leaf? This mesophyll can be divided into spongy as well as palisade. In the case of monocot leaf or dicot leaf? Quickly in the chat, students. Quickly in the chat, tell me. Monocot leaf or dicot leaf? Quickly. Students, I'll tell you the importance of this chapter. I will try to connect this chapter to every other chapter and teach you so you can have an entire whole revision. Okay? Or do you want me to only teach the chapter? Tell me, students. Do you want me to interconnect the chapters? Tell me yes or no. Yeah, exactly. Dicot leaf. Nice. Crazy all of you are. Okay. Columnar epithelial cells. Okay. Now, these are different sizes given here again. Sizes are given here. Okay. Students, this virus has reminded me something called as prions. Right? There's something called as prions. Do you think, students, prions, do they have a protein coat or only RNA? Do they have only genetic material or only protein coat? Tell me that in the chat, students. Tell me that in the chat. Quickly. This prion, not viruses, prions, do you think they only have genetic material or only have a protein coat? In the chat. Now, students, now we come to the prokaryotic cell in general. Prokaryotic cell in general right now. Now, where can you find prokaryotic cell? For example, in the case of bacteria, we can see prokaryotic, blue-green algae, mycoplasma, PPLO. Mostly kingdom Monera. You can see mostly kingdom Monera can be found here. Okay, kingdom Monera we can find here. Kingdom Monera. Okay. Now, students, see some of you are going wrong. See, students, when I ask you question, if you know the answer, type the answer. If you're doubtful somewhere, right? Sir, it's like, sir, I'm not able to remember it. That time, what you need to do is go open your NCERT and right now also in middle of the class, go look at it and come back. Okay, all of you will do that, students. All of you will do that. If you're not able to understand something, go and find right now the answer. Okay. Now, students, next, what do you have in the case of your prokaryotic cell? In the case of prokaryotic cell, students, the cell wall is made up of peptidoglycan here. Peptidoglycan right now. Now, I'll ask one more question here. Is peptidoglycan homopolymer, heteropolymer? Quickly, is peptidoglycan homopolymer, heteropolymer? Yesterday's class, biomolecules. Your peptidoglycan right here is, it is going to be heteropolysaccharide. Why? Because it is made up of NAG and NAM. NAG and NAM. Okay, NAG and NAM. Okay, peptidoglycan. In the case of mycoplasma, no cell wall. You have to remember that. You have to remember that, students. Okay. Now, cytoplasm is here. No well-defined nucleus. Obviously, ribosome is going to be 70S ribosome here. 70S ribosome. Okay. Genetic material is present here. Double-stranded, single or circular DNA can be present here. Double-stranded, single or circular DNA can be found here. Okay, heteropolysaccharide. Students, is chitin homo or hetero? Is chitin homopolysaccharide or heteropolysaccharide? Quickly, students, in the chat. Is chitin homo or heteropolysaccharide? Chitin is going to be homopolysaccharide. Remember that. Okay, remember that. I told you yesterday also, students, multiple times I told you chitin also. Remember chitin. It's homopolysaccharide. Okay. Now, there are different shapes are there. There is cocci, vibrio, bacilli, and spiral. Cocci is spherical, bacilli is rod shape, vibrio is comma shape, spiral is spiral shape. Spiral shape here. Okay, spiral shape, students. Now we come to something called as nuclear envelope and its modification. Ready, all of you? Ready, students, in the chat? Can I get a fire emoji if you're able to answer the question? Can I also tell students if you can like the video? Yes. Can we start? Students, can we start now? Cell envelope and its modification. Cell envelope and its modifications here. Okay. Now, first things first. Let's draw the entire cell structure right now. This is the second layer. All of you open your book. Okay. Done, students. Done. Now here, this is a typical structure of a bacteria. Let's say this is a bacteria right now. This is a bacteria right now here. Bacteria. Okay. Now, students, here this entire bacteria has 3 layers. First, outermost, second, third layer. 3 layers are present in this bacteria. Okay. Now, this is the outermost layer here. Outermost layer. Can anyone tell me what is the outermost layer called as, students? All of you. Something is itching, students. Anyone? Outermost layer is called as glycocalyx here. Glycocalyx. Now, Dinesh, understand. This glycocalyx here, students, can be of two ways. This glycocalyx can be present in two different ways. Okay. This glycocalyx can be present as loose sheath, or it can be a tough layer. Tough layer. Okay. When it is present as loose sheet, it is called as what? Slime layer. Slime layer. When it is present as tough layer, it is called as what, students? It is called as capsule. It is called as what? Capsule. Okay, students? Capsule. Now, second layer you understand. Okay, Christina, tell me what is second layer? Second layer is going to be what? Second layer is going to be cell wall here. Cell wall is going to be the second layer. What is cell wall made up of? Cell wall is made up of what, students? Who can tell me? Sarvanan, tell me. Cell wall is made up of peptidoglycan. Peptidoglycan. That is the second layer. Done, students. Then, which is the third most layer? Who can tell me? Saura, tell me, what is the third layer? Innermost layer is called as the cell membrane. It is called as what? Cell membrane, innermost layer. Innermost layer. Okay. In the right, in the center, what do we have? We have the cytoplasm here. We have the cytoplasm right in the middle. Cytoplasm right in the middle. Now, students, this bacteria, do you think should we make it motile? Devi, do you think should make it motile or non-motile? We'll make it motile. So if it has to show motility, does it need a separate legs-like structure? Does it need a tail here, a Hanuman tail here? Yes, sir, it needs. So we will draw on tail here. Okay, this tail is called as flagella. It is called as flagella right here. Okay, flagella done, students. Flagella done. Flagella done. Okay, flagella done here. Now, students, this particular bacteria, sometimes it has to attach to substratum. For that, we have one more structure here for the attachment to the substratum, that is called as pile. That is called as what, students? Pile, this one, for attachment purposes. This is called as what, students? Pile or fimbriae. Pile or fimbriae right here. Done. Now, apart from that, students, what do we have here? Do you think it has ribosomes? Yes, it has ribosomes here. So right in the middle, I will draw some ribosomes here. Okay. Now here, this ribosome here, students, is it 70S or 80S? All of you will tell me, sir, this ribosome in the middle is 70S ribosome. 70S ribosome is present here. 70S ribosome is present right in the middle. Okay. Now, students, does it have genetic material? Yes, it has genetic material. But do we have nucleus here? We don't have nucleus here. So all the genetic material is present as such here. All the genetic material is present as such here. Okay. And this genetic material right here, this genetic material is called as nucleoid. It is called as nucleoid. Nucleoid. Okay. Nucleoid. Then what is missing, students? What is missing? Now, from this plasma membrane, from this entire plasma membrane, there can be certain extensions like this. From this plasma membrane, right? From this plasma membrane, there can be certain extensions. Extension of plasma membrane right here. All of you can see. This extension right here. This extension of plasma membrane is called as mesosome. Mesosome. Okay, extension is called as what, students? Mesosome is right here, mesosome. Okay, now students, what happens is there are certain extensions here. There are certain extensions here in the cytoplasm. There are certain extensions here present in the cytoplasm from the cell membrane which has color in it, which has pigment in it. They are called as chromatophore. They are called as what? Chromatophore. They are called as what, students? Chromatophore right here. Chromatophore right here, students. Chromatophore. Okay. Chromatophore present here. Do they have vacuoles? Yes, they can be certain vacuoles can be present here. Vacuoles can be present here. There can be certain inclusion bodies somewhere. Inclusion bodies. Okay. Inclusion body for storage. So all of this is the entire cell envelope and its modification in the case of a bacteria. Done, students? Done? All of you? Done, students? Inclusion body. So I'll just revise this one more time for all of you. Right. Glycocalyx is the outermost layer. Loose slime layer. Tough, it is going to be capsule. Then second layer is called a cell wall, made up of your peptidoglycan. Innermost layer is called a cell membrane or plasma membrane, right, which is selectively permeable in nature. Inner part is called as the cytoplasm here. Here we have the ribosome, which is 70S. We have inclusion bodies here. Nucleoid is the genetic material here. Then we can also have a plasmid somewhere. Plasmid. Extra chromosomal self-replicating member called as plasmid can also be there. Yes. Then there is extension of plasma membrane. Extension of plasma membrane is called as mesosome. I'll tell you function of mesosome there. Then there is something called as a colored pigment called as colored type of pocket called as chromatophore, which is found in the case of blue-green algae. It is found in the case of blue-green algae. Blue-green algae. Student, this blue-green algae belongs to kingdom Plantae. True or false? This blue-green algae belongs to kingdom Plantae. True or false? Tell me. This blue-green algae belongs to kingdom Plantae. True or false? Yes. Yeah, we're done. Okay. Now, what is this mesosomes now? What are these mesosomes right now? Mesosomes. Okay. Mesosomes, I told you, they are extension of plasma membrane. They can be in the form of vesicle, tubular, or lamellar shape. All is possible. Okay. Now, what are the functions here? Cell wall formation. See, very important. This is where they ask you PYQ. The function of mesosome is called as cell wall formation, DNA replication, respiration, distribution of daughter cells, enzymatic content, increase in surface area, and secretion process. So I'll give you exactly 10 seconds to look at this and try to remember it, right? If you cannot, I will tell you one more time. All of you. Quick, students. Try to remember the functions now. I'll give you 10 seconds. I'll give you 10 seconds, students. 10 seconds, that's all. Done? Done, students? Okay. So that is the mesosomes. Function of mesosomes done. Okay. Now, what is peptidoglycan here, students? I told you peptidoglycan is the part of your cell wall. Now, depending upon the concentration of peptidoglycan, we can have gram-positive bacteria and gram-negative bacteria. And you can see here clearly that in the case of the gram-positive bacteria, large peptidoglycan is present. In the case of gram-negative bacteria, peptidoglycan is very less here. Okay, so we have gram-positive and gram-negative bacteria can be present here. Okay, now what is flagella? Okay, what is flagella right now? Yes, flagella is nothing but, students, I told you, if a bacteria has to move, it needs some kind of tail. With the help of the tail, it is moving. The tail here is called as flagella here. Okay, and flagella here, students, is very, very simple compared to eukaryotes. Okay, here flagella is so simple. Flagella is so simple. It has only 3 parts. Here, can all of you see? The first part is called as basal body. Second part is called as what, students? First part is called as basal body. Second part is called as hook. Hook. And then the inner part, this part is called as filament. It's called as filament. So here 3 parts are there. Basal body, hook, and filament are the 3 parts of the flagella. Done, students? Done. Okay. Basal body who can filament. Can all students see this? From plasma membrane, it is coming out. It is coming out here. It's coming out like this. This is the basal body, hook and filament. Hook and filament. Now we come to ribosomes again, students. Ribosomes. Now here ribosome, I told you it's 70th ribosome, and the 70th ribosome is divided into two categories. It is divided into something called as your 50s and 30s. Okay, large subunit, small subunit. When these two subunits join together, we get the entire ribosome. Now here, students, what is the S here? Can anyone tell me, students, what is SS here? Who can tell me? What is S here, students? Sedimentation coefficient, correct. Sedberg unit, correct. So the S here is nothing but sedimentation coefficient, or it can also be called as Sedberg unit. It can also be called as what? Sedberg unit. This one, S here. S. Sedimentation coefficient or Sedberg unit. Okay, students? All of you? Done? Done? Alright, otherwise you'll forget it off. Sedimentation coefficient, or it can be called as Sedberg's unit. Sedberg's unit can also be called as, that is the S here. Okay, S here. Now again, let me ask all of you on something. This S, right, this ribosome, students, do you think it has a membrane around it or not? Do you think it has a membrane around it? No. Ribosomes do not have any membrane around it. There's no membrane around them, ribosomes. Okay. Now, then there's something called as polyribosome. What is polyribosome? Poly means what? Poly means many. Ribosome means ribosome. So, many ribosomes is called as polyribosome. And this happens on a single mRNA. So, if on a single mRNA, if many ribosomes are present, several ribosomes plus single mRNA, that is called as polysome or polyribosome or polyribosome. Okay, no membrane. Surya, no membrane. Okay, now there is something called as inclusion body. What is inclusion body, students? Inclusion body is the storage unit in the case of your bacteria. So all the waste or unwanted things or anything is always stored in the case of the inclusion body. You include it there. You included there, students. Included there. Okay, it is free in the cytoplasm. Obviously, no membrane at all. Examples are there. Phosphate granules is an example. Cyanophysin granules is an example. Glycogen granules. See, in glycogen granules, glycogen is stored. Then there is something called as gas vacuoles. Gas vacuoles are found in the case of purple and green sulfur bacteria. In purple and green sulfur bacteria, we can find gas vacuoles can be found here. Okay, gas vacuoles can be found here. Okay. Now, students, here is the question on the board. Let's see who can tell the answer. Who can tell the answer? Cell wall formation in bacteria is facilitated by cell wall formation. Who helps in cell wall formation? Golgi? No. There's no Golgi. There's no centrosome. Ribosome? No. I told you, students. Mesosome helps in what? Mesosome helps in cell wall formation. Yes, bold, Desh. Okay. It is going to be mesosome. This is the NEET 2024 question. Last year question. Okay, look at this question, students. Look at this question, all of you. Inclusion bodies of blue, green, purple, and green photosynthetic bacteria are inclusion bodies here. See, if they would have given you cyanophysin granules, if they would have given you glycogen granules, right, then it would have been a little confusing. But it's so easy now, students. It's so easy now. You can tell me very easily. I have not even thought you conjugal vacuole, centrioles, microtubules. No. It is going to be gas vacuoles. Gas vacuoles are present here. Gas vacuoles are present in the case of your blue-green and purple-green sulfur bacteria. Now, this blue-green and sulfur-green bacteria, students, did we study them in the chapter photosynthesis and higher plants? Tell me yes or no. Which scientist was working on them? Which scientist was working on them, students? Who can tell me? Which scientist was working on he was a microbiologist. He told us that water is the electron, water is the hydrogen donor. Yes, water is the hydrogen donor here. He also told us that in purple and green sulfur bacteria, H2S is released. Here, H2S is released here instead of the, and here sulfur is released. Sulfur is released, sorry, H2S is used. Sulfur is released as a byproduct here. Sulfur is released as a byproduct here, right? Sulfur, not TW Engelmann, no, 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 not TW Engelmann. TW Engelmann was working on what? TW Engelmann was working on what? He was working on aerobic bacteria. He was working on aerobic bacteria, students. TW Engelmann was working on aerobic bacteria. Tell me, students, which scientist told us about that hydrogen is the, sorry, water is the hydrogen donor? Exactly, Cornelius Van Niel experiment. Cornelius Van Niel experiment, students. Okay. Students, should I ask you more questions now again? Tell me. Huh? Should I ask you more questions now? What is this? Okay, tell me, students. Who discovered oxygen? Who discovered oxygen? Joseph Priestley. Joseph Priestley. Tell me now, quickly, students. In which step of your Kelvin cycle 1 ATP is consumed? In which step of Kelvin cycle 1 ATP is consumed? Quickly answer. Quick answer I want. After you answer that, you answer this question also. This question, quickly. What is this? Plant physiology again gone already. Huh? Plant physiology out of brain removed already. What is this? Reduction, exactly. So, students, reduction or regeneration? Students, the question was what? In which stage of Kelvin cycle 1 ATP is consumed? In reduction, 2 ATP is consumed. In regeneration, 1 ATP is consumed. All of you are wrong. Huh? All of you are wrong. It is regeneration. Students, should we conduct a plant physiology marathon after this? Should we conduct a plant physiology marathon? Huh? Should we? Do you want one shot plant physiology we will do in next coming days? It is on reduction, regeneration. In reduction, 2 ATP is consumed. 2 ATP is consumed in reduction as well as 2 NADPH is consumed. What is this? The concept of Omnis cellula-e-cellula regarding cell division was first proposed by Rudolf Virchow. Very easy. Okay. Now, if this is over, now can we go to the next, students? Can we start eukaryotic cell now? Huh? Can we start eukaryotic cell right now, students? All of you. Can I get some fire emoji in the chat? All of you. If I keep asking questions, all of you be like, sir, please be, sir, like, sir, enough, sir. We cannot answer anymore. Huh? Okay, tell me one more question before I go further. One more question. Oh God. Tell me, students, which is the primary electron acceptor in the case of cyclic photophosphorylation? This is my favorite question. This is my favorite question, students. Okay. Tell me, which is the primary electron acceptor in the case of cyclic photophosphorylation? Who can tell me? See this. If you're part of a crash course, I've told you recently in crash course. Tell me now. All of you do not want questions. You're like, sir, enough questions now. Tell me. Not pheophytin, students. Pheophytin, no. Pheophytin is going to be part of non-cyclic. It is iron sulfur centers. Beautiful, nice. Iron sulfur centers. Nice. Iron sulfur centers. Now all of you will answer now. Now all of you will answer. Okay. Now, in eukaryotic cell, where can we find? In Protista, plants, animals, and fungi. Now, who can tell me, students, what mode of right nutrition is found in the case of fungi? Option number one, saprophytic mode of nutrition. Option number 2, parasitic mode of nutrition. Option number 3, mutualistic relationship. Option number 4, all of the above. Tell me the answer. Tell me the answer. Quickly. Which mode of nutrition is found in the case of kingdom fungi? Saprophytic, parasitic, mutualistic, or all of the above? Answer is all of the above. The answer, see, the students who are saying all of the above, recently I taught them this chapter in your crash course. Yes or no? Tell me now, isn't it beneficial now? If you did not learn from me, you will tell saprophytic, sir, saprophytic. No, it is not. We get Puccinia, albugo, parasitic mode of nutrition, right? Mycorrhizae, lichen, mutualistic relationship. So answer is all of the above. What, students? Huh? I'll ask you one more question now. Wait then. Huh? Tell me, students. Amoeba. No. Sleeping sickness is part of. Sleeping sickness is caused by which organism? Sleeping sickness. Quickly, students. Tell me, students. Sleeping sickness is caused by which? Sleeping sickness is caused by which organism? Sleeping sickness. Right? And they belong to which kingdom also? Tell me. And which belongs to which kingdom? Trypanosoma. Yes. Which kingdom? Which kingdom? Quickly. Protista. Flagellated protozoan. Exactly. Beautiful. Beautiful. Okay. Okay. Tell me your okay. Nice question. This is a nice question. Formation of plasmodium is observed in which organism? Formation of plasmodium is observed in which organism, students? Quickly in the chat. Under favorable condition, this organism will make plasmodium, and this plasmodium can undergo form buds and new organism can be found. Tell me, plasmodium. Who forms plasmodium? Tell me answer in the chat. I'll look later. Okay. Now, what are characteristics of eukaryotic cell? Membrane bound organelles. Yes, membrane organelles. Organized nucleus with nuclear envelope. Yes, it is found here. Complex locomotory and cytoskeletal structures are found here. DNA is double stranded and it is in the form of chromosome. Slime mold. Whoever told slime mold is genius right now. Students, this is where you go wrong. This is where you go wrong, student. This is where they catch hold of you. This is where they catch hold of you. I told you plasmodium is the organism. I told you which organism is found is making a structure called as plasmodium. I'll show you, students. All of you keep forgetting. No, this is where I catch hold of you, student. This is where not only me, your examination also catch holds of you. This is what happens when you don't revise. This is what happens if you're not a part of crash course. You will not tell. Okay. Students, see this here. Yappa. See here. See, sporozoans, plasmodium is an example. They are a parasite. But if you look under students, if you look under slime mold right now, it says the unfavorable conditions, right? See here. Under suitable condition, they form an aggregate called as plasmodium, which may grow and spread over several feet. So plasmodium, can all of you see? See this, zoom your eyes, all of you. Plasmodium. So it is going to be slime mold. Okay. Tell me, students. Understood? Understood, all of you? Yes. So students, if you get such questions, you're cooked. I'm telling you now only. Question will come from anywhere, and I'm telling you, students, this year you have to be prepared very well. You have to be so well prepared about your revision, is that if the question comes from anywhere, you should be like that. Okay? Like that. Okay, students? Now, let's see this. Now, plant cell versus animal cell. Who can tell me in which of the cells we find plastid? Tell me. See, there are two plasmodium. One plasmodium, I'm talking about plasmodium formation. One more plasmodium is a parasite. That is different. That is in sporozoan. Okay. That is sporozoan. Now tell me students, in plant cell, do we get plastid or not? Yes sir, here we find plastids. In the case of animal cell, no plastid. Plastid. Here we find cell wall. Here cell wall is absent. Okay. Now students, here we find centrosome. Centrosome is present or absent here? Centrosome is absent here. Okay. Large nucleus, sorry, large vacuole. Where do you find large vacuole, students? Large central vacuole, right? Large central vacuole is found here, not found here. It is found here, but it is very, very small. Very small here. Okay. Tell me here, students, which is the plant cell here? Is A plant cell or B plant cell? Quickly, students. Which is the plant cell here? Which is the plant cell here? Quickly. A is going to be the plant cell. We can find chloroplast here. We can find the chloroplast here. Now we come to cell membrane or plasma membrane. Now we come to cell membrane or plasma membrane right now. Can we start? Can we start, students? Again, students, tell me in the chat right now. Do you honestly want more questions from me or do you want me to only teach and go? Tell me, students. See, I can do two things right now. I can just teach you this chapter and I can go, or I can connect this chapter to every other chapter and ask you as many questions as possible to make you revise the entire concept of every other chapter, and you will feel much more better. You will feel much more better, let me tell you that. Because when I ask you a question, if you do not know the answer, you will go find it later on at least. And learning becomes double the learning it happens. Okay, double the learning it happens. Okay. Now, what is the cell membrane or plasma made up of? It is made up of lipids. Yes, lipids, protein, carbohydrates. It is made up of lipids. It has proteins. It has carbs, carbohydrates. It has water. It has the cholesterol. It also has the cholesterol. It also has the cholesterol. Cholesterol here. Okay. It also has the cholesterol here. Okay. Now, students, this entire cell membrane. Okay. Entire cell membrane, students, is it living or dead? Tell me. It is going to be living in nature. Okay. It is going to be asymmetrical in nature. Asymmetrical. Now, students, tell me in the chat. This is going to be, let's say, it is going to be mosaic in nature. It is going to be mosaic. Meaning of mosaic is random. It is going to be random in nature. Now, students, again, I've always told you, students, who is the inspiration? Who is the inspiration for a cell membrane? Cell membrane came into existence by looking at this. They're like, wait a minute, this is a nice model. This is a nice model. I will copy this and I'll become like that. Who tell me, students? Who is the inspiration? Who is the inspiration for your cell membrane? Quickly. Quickly, students, quickly. The inspiration for a cell membrane is none other than, who can tell me, students, quickly? The inspiration of cell membrane is none other than our Indian roads. Tada! Like Indian roads, what happens? In Indian roads, you are going nicely. You think you are going, you're singing a song, thinking about someone, you're going. Do. Suddenly one pothole will come. Suddenly one hump will come. Pothole hump. And ultimately you'll go fall somewhere. Okay. So cell membrane is also like that. Your entire cell membrane is also like that. It is very random. I'll show you a diagram, students. See this. It is going to be completely random. See this. How random is this? You're going randomly on hump. Hump is coming from nowhere. Hump, one more hump. Okay. So it is going to be our reality, students. Reality. So that is the inspiration. That is the inspiration right here. Okay. Now, that is the meaning of asymmetrical. Okay. Who can tell me, students? What is the meaning of mosaic? Mosaic means, students. No, student. Today, dad jokes. Yesterday, I got some nice. All of you did not like my dad jokes, so I will not tell dad jokes anymore. Okay. Okay. Do you want to hear one joke though? I can't resist myself. Do you want to hear one joke? Okay, listen. Why do you think, okay, why do you think there are so many different types of flowers in the world? Anyone can tell me. Why do you think there are, students, so many different types of flowers in the world? Why? Why, students? Because every flower has its own style. That's why. Every flower has its own style. That's why. It's a botany dad joke now. Okay, students. Next question here. Okay. So meaning of mosaic is what? Mosaic is this one. This is called as mosaic floor. This is called as mosaic floor right now. Mosaic floor. Okay. Now, if you see, everything is so random here. Everything is so random. Something all is going on. Something all is going on here. Okay. So it is so random. So your entire cell membrane is also very random. Cell membrane also is very random. Okay. Cell wall is also very random here. Okay, students. Okay. Done. Students, your entire cell membrane is said to be semi-fluid in nature. It is also called as quasi-fluid. Quasi-fluid. Now, what is the meaning of quasi-fluid? You understand. Why is it called as quasi-fluid, or is it called as semi-fluid in nature or semi-solid in nature? Okay. Now, this particular cell membrane has both the properties. It has both the properties. It can be tough also, but it also has a property of liquid where things can move in it. Can you move in solid? You cannot move in solid. Okay. But, okay. Can it move in liquid? We can move in liquid. So things can move in the cell membrane. Things can move in the cell membrane. That is why it's called as semi-liquid or semi-solid or what? Quasi-fluid in nature. Okay. Because it has solid-like property and liquid-like property. Half and half. Not here also, not there also. Somewhere in the middle. Somewhere in the middle. Okay. Now, students. Now, what is cell membrane actually made up of? Right. The cell membrane is actually made up of something called as the lipid bilayer. Bi means what? Two. Lipid, two layers of lipid. Now, in these two layers of lipid, students, the head region is called as the polar head, which is hydrophilic in nature. Philic means what, students? Philic means loving. It loves full. It is loving in nature. Now, what is non-polar tail means? Hydrophobic in nature. Like, go away from here. Go away. Okay. So, hydrophobic means it is scared of water. Okay. Scared of water. Okay. This right here is a phospholipid bilayer. Okay. Phospholipid bilayer. Now, again, students, who can remind me which lipid can be found here? Okay. Why does the dosa fly? I don't know, students. Why does dosa fly? You should tell me why does the dosa fly? Tell me, students, quickly in the chat. Which type of lipid do we find here? Which type of lipid? It is a type of your compound lipid. Quickly. Who can tell me? It has a phosphate in it. Lecithin. Beautiful. Paper dosa. Nice, nice. It is called lecithin. It is called, students, what? Lecithin. Okay, lecithin. Cool. Phospholipid bilayer, students. Phospholipid bilayer. Can all of you see the arrangement here? All the head is towards the outside. Tail is present in the middle here. Tail is present in the middle. Okay. Now, this is how it can look like. This is how it can look like, students. Entire phospholipid bilayer. Phospholipid bilayer. Okay, students. Now we have one more model here, students. This is called as your fluid mosaic model. This is called as, students, this is called as fluid mosaic model. Fluid mosaic model right here. Fluid mosaic model. This fluid mosaic model was given by Singer and Nicholson in the year 1972. By Singer and Nicholson in 1972 by Singer and Nicholson. This is called as fluid mosaic model. Okay, it's called as fluid mosaic model given here. Now, students, you tell me here. Here, do you see any proteins here, students? Do you see any proteins here? Tell me in the chat, tell me yes or no. Do you see proteins here? I can see proteins here. Now, students, here, understand. I will erase this, all of you remember this, okay? Singer Nicholson. Here, I can find two types of proteins here. I can see some proteins are present on the outside, some proteins are present on the inside. Now, based on the ease of extraction, what do I mean by that? For example, students, I have nothing to show you here, students. There's nothing here. Okay, I'll try to take this. Okay. Now see this, students. Imagine this is your lipid bilayer. Okay, and I am keeping the protein, or one protein is somewhere in the inside like this. Let's say the protein is almost inside. Now tell me, students, is it easy to remove? Difficult to remove? It's not coming out. Like, not coming out now. Difficult to remove. Difficult to remove. Now what if I keep a pen this much on top like this? Now tell me, students. Now I can easily hold and remove. Based on the ease of extraction, how easy is it to remove something? We have two types of proteins here called as integral protein, integral proteins, as well as the peripheral proteins. Peripheral proteins. Integral proteins are present on the inside. Integral proteins are also called as transmembrane proteins. Transmembrane protein. These transmembrane proteins are present on the inside. They're present on the inside here. This is the integral. Okay, integral proteins. Now peripheral is present on the outside, also called as extrinsic. Also called as what, students? Extrinsic protein. The outer part, extrinsic protein. Okay. Pen and tissue paper. Yes, there's no, see, that was in the house. Here nothing here, students. I cannot even show you. But all of you understanding, right? All of you understanding. Okay. Now, students. Now we come to the movement here. Understand the movement. Okay. In the case of cell membrane, plasma membrane, there are two types of movements here. One is called as flip-flop movement. Other one is called as lateral movement. Now, what is flip-flop? Understand, all of you. Understand. Let's take a lipid bilayer here. Okay. Here we have a lipid bilayer right here. Lipid bilayer present right here. Okay. Now, what is flip-flop? Flip-flop is nothing but if this lipid right here, lipid right here, if it comes this side and this one goes this side, they are doing exchange offer. That is called as flip-flop movement. That is called as, students, what? Flip-flop movement, students. Flip-flop movement. Okay. Flip-flop movement now. Okay. Now understand. Not flipkart, flip-flop. Flipkart, it seems. Now, what is lateral movement? For example, right in the middle, I have a protein here. Let's say I have a protein here. Now, students, can this protein right now undergo flip-flop? Not possible. If it undergoes flip-flop, it will go somewhere out of the picture only. Okay. So, this protein right here, this protein right in the middle right now, this protein right now will show something called as lateral movement now. So, this will move laterally now. If this, let's say I am the protein, I'll go like this. This is called as what? Lateral movement. This is called as what, students? Lateral movement. Moving sideways, students. Okay. Okay, students. Understood? That is lateral movement shown by here. Flip-flop is shifting up and down. What are the functions here? Movement of proteins, endocytosis, cell division, secretion, formation of intercellular junctions. All of these are different functions of the cell membrane. Okay, cell membrane, students. Now there is something called as active transport and passive transport. Understand what is active transport, what is passive transport. Okay, active transport means what? Passive means what? No energy is required along the concentration gradient. It happens. This I'll give you a simple example. If you have traveled in any government bus, if you travel in Bangalore via BMTC, in the peak time, if you go stand near the BMTC, right, what will happen? You don't even have to climb the bus. Within 2 minutes, you will automatically go inside the bus. All you have to do is, stand in the line. Okay. If you stand in the line, people will push you automatically without any energy. You will be sitting inside the bus next minute. Okay. That is called as, students, that is called as what? Passive transport. You might have observed passive transport even in the case of your temple also. Students, if you have gone to Tirupati, okay, you know if you have gone to Tirupati, right? If you stand in a line, what happens? Next, even if you don't move, they'll make you move. They'll make you move in Tirupati. Okay. In any recently I went to this temple. Okay, recently when you go, I went to temple, I forgot the name of the temple. I am standing. I spent around 15-20 minutes waiting in the line in the temple. By the time I went to see the God is there. I am standing here. Before I could do a namaskar and do this, I am pushed out of the frame. Next thing I am seeing, I am doing namaskar to a pillar there. I am doing a namaskar to a pillar there. Okay. So next level it is pushing. That is called as passive transport. You don't have to do anything there. Automatically you will move. Okay. Now, next one is called as active transport. In active transport, you have to move on your own. Okay, you have to do on your own. It's like waking up early morning from the bed. Early morning from the bed, very difficult to move. Very difficult. You're like, I want to sleep for 10 more minutes. Okay. In such case, what happens? You have to use energy. Use ATP here. Okay. So, and active transport, right? Active transport, polar molecules cannot pass through usually. Polar molecules cannot pass through usually. Okay. So, what happens is, this polar molecule will call a cab. Bro, I cannot go from here to here. You come and pick me up. Like, Macha, you come and pick me up. So, this polar molecule will be picked up by a carrier protein, and this carrier protein will take them inside. This carrier protein will take them inside. That is all it's called as active transport. Here it happens against the concentration gradient. From low to high it goes. Low concentration to high concentration. Okay. Low concentration to high concentration. Example is sodium potassium pump here. Okay. Here it goes from high to low along the concentration gradient. Example is osmosis here, where water movement happens. All of you understood, students? All of you understood? All of you understood or not? Tell me. Yes or no? Passive and active transport. Yes? Understood? No. This aquaporins no need. Now we come to something called as cell wall here. Now we come to something called as cell wall. Okay, cell wall. Cell wall is very special. Cell wall is very, very special here. Okay. Now, unlike the plasma membrane or cell membrane, cell wall is dead. Unfortunately, students, cell wall is dead here. Cell wall is dead here. Okay. And students, can anyone tell me, students, is cell wall present outside the cell membrane or inside the cell membrane? For example, students, this is a cell membrane right here. This is a cell membrane right here. Now, cell wall will be present always. Cell wall will be present always on the outer wall here. This is the cell wall present on the outer side of the plasma membrane. Outer side of the plasma membrane, it is present on the outside. Okay, present on the outside, students. Okay, it is going to be non-living in nature. It is going to be outer layer. It is going to be outer layer, right? We can find in the case of your plants. We can find in the case of fungi. We can find them in the case of certain protists also. Now, can anyone tell me, students, the cell wall in the case of fungi is made up of what? Students, I need water. There is no water here. Tell me, students, quickly in the chat. Animals are not present, yes. Fungi, what is cell wall made up of? In the case of fungi, the entire cell wall is made up of chitin. Chitin and polysaccharides. Remember that. Chitin and polysaccharides. Remember that, students. Remember that. Now, what are the functions here? Function. Are there any function? Yes, there is function. Function of the cell wall is protection. Protection. It is like a barrier. It is like a barrier here. It prevents cell from going mental. That is bursting. Okay. Prevents cell from bursting. It counteracts osmotic pressure. Counters osmotic pressure. Osmotic pressure is counted here. Okay. It is counted. Yes. Shape and rigidity also. Shape and rigidity also provides cell wall. Now, students, there are what is in case of algae? In the case of algae, it is made of cellulose, galactans, mannans, calcium carbonate. In the case of plants, it is made up of cellulose, hemicellulose, and pectin. In the case of fungi, it is made up of chitin. This you have to remember. This you have to remember, students. Okay. I'm thinking, what can I ask you a question here? Cellulose. Okay. Tell me, students, can cellulose hold iodine? Can the cellulose hold iodine, students? This is cell the unit of life, students. This is cell the unit of life. Okay. This cell the unit of life. What is that? Structural organization animals, Gopika Ma'am will come and teach. Okay. Tell me, students, cellulose, can it hold iodine or not? No, it cannot hold iodine. Or yes, it can hold iodine. Remember the structure of cellulose. Structure of cellulose. You remember, students, cellulose? Starch can hold. Starch can hold. Because starch has something called as helicals. Starch has something called as helicals. Okay. Now, what about in the case of cellulose? Can it hold iodine? It cannot. Cellulose does not have helicals. Cellulose is a beta 1,4 linkage. It is in a straight chain. All of you forgot, no, students? All of you forgot, right? All of you forgot, no? Yesterday only I taught you, students. Yesterday I taught you. It does not form complex helicals. Does not form complex helicals, so it cannot hold. It cannot hold. All of you like, sir, don't ask questions anymore. Now, how is a cell wall made? Okay, how is cell wall made? Here you can see, students, there is something called as primary cell wall on the outside. Primary cell wall on the outside. It is present in young plants. It is permeable and can show growth. Right? I can show growth. On the inside, from the out, this is a primary cell wall. On the inside, what happens? Secondary cell wall formation happens. Non-elastic, non-permeable. Why? Because of suberin ligand deposition here. Now, tell me, students, in the chat, what if I make tertiary cell wall now? Tertiary cell wall. Will the tertiary cell wall come on the inside or outside? Tell me. If I make a tertiary cell wall, it will come on the inside. This is going to be tertiary cell wall. Tertiary cell wall right here. Okay. You can see, students, primary, second, tertiary, it's forming on the inside. Tertiary is forming on the inside here. Inside. Okay. Now understand, students, next thing. Now, if this is one cell here and one cell here, between the two cells, there is a gum here. There is a glue here. Okay. Fevicol. The middle part here, between the two cells, is called as middle lamellae. It's called as what? Middle lamella also. Middle lamella. Right in the middle, there is middle lamella between the two cells, right in the middle. Middle lamella here. Now, what is the difference between middle lamella? What is the difference between middle lamella as well as plasmodesmata? Small difference, understand. Small difference, understand, students. Okay. Here, plasmodesmata is nothing but, right, the cytoplasm here. The cytoplasm of this cell and cytoplasm of this cell, both the cytoplasms are interconnected here. Can you see? So plasmodesmata is nothing but, it is a cytoplasmic connection. It is nothing but a cytoplasmic connection between the two cells here. This right here. Cytoplasmic connection between the two cells. That is called as plasmodesmata. Okay. Cytoplasmodesmata. Students, I can't show animation on YouTube because, you know, copyright strike will come. Okay. Now, if that is clear, students, now we come to something called as endomembrane system. Can we start? Can we start, students? Can we start? Students, give me a minute. Let me just go get some water. Give me a minute. I'll go get some water, students. I'm feeling very thirsty. Give me a minute. Now, students, now we learn something called as endomembrane system, students. Endomembrane system. Okay. Now, in endomembrane system, how will the question come? In endomembrane system, they can generally ask you. In endomembrane system, they can generally ask you what are the organelles present here and why are they present together. That's all. Now, here is a small trick to all of you. The small trick here is, students, every gamer loves victory. So every gamer loves victory. The small trick here. Okay, small trick right here. Now, what is E? E stands for endoplasmic reticulum. ER. G stands for Golgi complex. L stands for lysosomes. Okay. V stands for vacuole. This right here, student. This right here is the endomembrane system. All four of them. Now, all four of the students, why are they present together? Because all of their functions are coordinated. If you learn this much, you can easily get 4 marks. Students. Okay? Easily get 4 marks. Okay? Yeah, we can ask Gopika Ma'am once. Okay. Now, let's see a question here, students. Let's see a question. Tell me the answer to this question, students. Can anyone tell me the answer? Quick. See, membrane-bound organelles of endomembrane system coordinate cellular functions. Okay. Mitochondria and chloroplast are not considered a part of endomembrane system. Obviously, mitochondria and chloroplast are not a part of it. Look at this question now. The organelles that are included in the endomembrane system are, which of them are part of endomembrane system? Okay. Quick, students, quick. You don't have to write don't worry. Students, you don't have to write the notes. I'll give you the notes. Okay, don't worry. I'll just scribble some notes right now. Do not worry, I will share you the PDF. You can download and read from there. Be happy after that. Okay, don't worry. Quick. See, mitochondria is there? Not gone. Mitochondria gone. So what is the answer, students? What is the answer? Endoplasmic reticulum, Golgi complex, lysosome, vacuole. All four of them are part of endomembrane system. They're part of what? Endomembrane system, students. Endomembrane system. Okay. The first one is called as endoplasmic reticulum, also called as ER. Endoplasmic reticulum, also called as the ER, students. Now, what exactly is ER? Understand all of you. What exactly is the ER? ER is nothing, students. It is a network or reticulate of tiny tube-like structures. Reticulate tiny tube-like structures. Okay. For example, students, let's take one cell here. Let's say this is a cell right now. This is one cell right now. Okay. In this cell, students, right in the middle, we have the nucleus. Let's say we have nucleus here. Now, what happens is, from the outer membrane of the nucleus, nuclear envelope is there. From the outer membrane, see, there are two layers here. All of you should be aware that there are 2 layers for a nucleus. From the outer layer, we can see the development of ER. We can see the development of ER from the outer wall here. From the outer wall here. Okay. Now, students, understand here. Whatever is present inside this tube-like structure here, this one, the entire tube-like structure right here, this region is also called as luminal region. Luminal region. Luminal. Now, whatever is present on the outside here, this region, where we do not have the tubes, in this cell, where the tubes are not present here, this outer part, this region is called as what? Extra luminal. It is called what, students? Extra luminal. So luminal as well as extra luminal is found here, students. Luminal as well as extra luminal is found here. Okay. Luminal as well as extra luminal. Now this is a extension of plasma membrane. Okay, external plasma membrane. You can see, students, here the ER is dividing the entire cell. The ER is dividing the entire cell into two parts, luminal as well as extra luminal. Two parts. Okay, two parts here. Now, students, understand here. Here, if ER has something called as ribosomes, then it becomes something called as RER. Then what if the ER has no ribosomes? Then it is called as SER. Now this RER can produce something called as proteins, involved in protein synthesis, while your SER is involved in the lipid synthesis. SER is involved in the lipid synthesis here. Okay. Okay, students. Oh. Nuclear envelope. Extension of nuclear membrane. Not plasma membrane, but nuclear membrane. Nuclear membrane, not plasma membrane, nuclear membrane. Okay, students. Nuclear envelope or nuclear membrane. The outer part. All of you know there's the inner and outer here. And the outer part becomes stick. Okay. Lipid synthesis it can make. That is your entire ER. SER and RER. Lipid synthesis as well as certain steroidal hormones this also it can do. While your RER produces proteins. RER produces proteins here. Okay. Can all of you see here? This is called as the nucleus. Nuclear envelope. From the nuclear envelope it is coming out. Nuclear envelope it is coming out here. Entire thing. Okay. Question on the board quickly. Quick answer I want, quickly. Which of the following statements concerning the endoplasmic reticulum is incorrect? See, this is correct. Correct. In prokaryotes, only RER is present, students? No. In the case of prokaryotes, ribosomes are only not present. They are not present at all. Okay. So in the case of your prokaryotes, rough endoplasmic reticulum is not present. Okay. Even SER is not present. RER is also not present here. Both are not present. Okay. Both are not present here. Okay, students. No. Students, how many of you keep ordering online? Can anyone tell me? How many of you keep on ordering online? How many of you keep on ordering online, students? You're like, sir, I don't have control over myself. I can keep ordering online shopping. I do a lot of online shopping. I keep ordering t-shirts and everything. Right? So when you order something online, does it come with a package or does it come without a package, students? Anyone who can tell me? Will it come with a package or without a package? Will they just throw the t-shirt and go at your face? No. What do? They do nicely do a package. It comes in a box and then give you the box. Similarly, students, inside a cell, there is one organelle with the main responsibility is packaging. Its main responsibility is packaging everything. And the organelle which I'm telling here is the organelle which I'm telling here is none other than the Golgi complex. It is going to be the Golgi complex. Golgi complex was, hello ma'am. I just got this, ma'am. No, I just rang it up. I just say hi to them, ma'am. Oh god. Exactly, Golgi. Golgi complex is the Amazon factory here. Okay, because all it does, it doesn't produce anything. A little bit of carbohydrates it can produce, but main work is here is packaging. Nicely, it'll package everything, it'll give it. They take it and go. Okay. It was discovered by Camillo Golgi right here. Let's say hi.
Hi. Hi guys, how is the Josh?
Yeah, ma'am. They are very demotivated, ma'am. I asked them a few difficult questions. They're like, oh my god.
It's okay. Part of this. Yes, how is it going? We have to do structural organization, no? I am very tired. Yes. Come on, I think the messages are loading very late, no?
Major lag is there.
Yeah, there's a lot of lag. So this is hot. 1 second, students. Yes. The mouse will come like this, sir. Hey, what do you drink? Coffee? Hello. Hi. Yes, GMA is very nice. Okay. So how long?
I'll take one more. Or less than that.
One more hour, sir? No.
You'll get over. I'm done with half the chapter is over almost. More than half is over. Okay. Right. After this, we have certain organelles, and then there is certain structures. That's all, which they don't understand usually.
Yeah. Okay. Let's hope it's not for an hour.
Ma'am, I started at 11:15, I guess. 11:15.
Okay. The thing is, they'll be exhausted.
Yeah. I will drain them.
Can't hold it anymore.
Yeah. Huh. Yeah. Then it got clicked. Okay. They thought I'm taking SAO. I thought I'm SAO. SOIA. Ma'am will come, students. Ma'am will come. I will not take SOIA. Okay. So students, let's start. All of you. Can we start now? Again. Now, students, can I get for the next 10 minutes ultimate attention? 5 minutes. Because if you don't understand this part, right, the concept of Golgi, if you read NCERT, it is very difficult to understand. So what I'll do is right now, I'll teach in the best way possible the entire packaging, how it is done. Okay. And then you can continue very well. Okay. Understand, students. Understand, all of you. Understand. Okay. There is something called as, students, here, flat disc-shaped sac-like structure called as cisternae here. Now I've told you, students, always I've told you a small trick here. The small trick here is cisternae, long word. Cristae and cisternae are two different things. Remember, cisternae is longer, Golgi apparatus is longer. Just remember that cisternae are found in Golgi apparatus. Okay. Now, here Golgi cisternae are arranged concentrically. They are arranged concentrically here. Now, students, understand here is that if this is the nucleus, this right here is the ER. This right here is the ER right now. Now, here we have the Golgi body. Here we have the entire Golgi body here. Now, students, understand here is that this entire Golgi, the entire Golgi body has two faces. Just like your some friends, they have two faces here. Okay. What are the two faces here? The first face is called as one which is towards the nucleus or towards the endoplasmic reticulum. That is called as your convex phase, cis phase, or forming phase. I'll repeat again, students. Understand. Towards the nucleus, right? Towards the nucleus, right? Towards nucleus, this phase is called as what? Forming phase, cis phase, or convex phase. This is convex phase. Okay. Now, one which is towards the cytoplasm, this phase right here, this phase right here is called as concave phase, trans phase, or maturing phase. Towards the cytoplasm, this is called as maturing phase. Now, who will produce something now, students? Who will produce something now? Here, ER is going to produce proteins. It can produce lipids. ER can produce proteins or it can produce lipids here. Now, these lipids and proteins, where are they entering first of all now? Understand here. See. Now, here we have cis phase. So, all the proteins All the proteins and lipids, they will enter from where? Cis phase. Entering from the cis phase, they're entering now. All the protein and lipids are entering from cis phase. Now, once it enters here, it undergoes packaging now. Packaging, until it is coming out of the trans phase. It is nicely packaged and exit is happening from the trans phase now. Exit is happening from the trans phase. So, all the packaging is happening right here. All the proteins or lipids produced by ER is undergoing packaging. Exiting from trans phase. Entry is cis phase or forming phase. Trans phase is maturing phase. It is coming out. So just learn the entire flow. You're good to go then. You're good to go. Clear all of you? Clear? See here also we can see. We have cis phase, it is entering. Trans phase exiting. Trans phase exiting is happening. Okay. And in trans phase we can see certain vesicle formation. Round vesicles can be seen here in trans phase where they're coming out. Okay. Okay, friends. Now, what are the functions of Golgi complex? The Golgi complex can mainly produce two things. It can produce something called as glycoprotein as well as glycolipids. Okay. In this glycoprotein and glycolipid, students, this glyco part is added by Golgi complex, while this lipid is given by SER. Again, glyco by the Golgi complex here, Golgi complex, and this protein is given by RER. So this protein and lipids are undergoing packaging now. They're undergoing packaging, and we get the product here. Okay, proteins. Students, can anyone tell me in protein structure, which structure is mostly bioactive in nature? Anyone who can tell me? Which protein structure is bioactive in nature, which can show 3D structures? Yes, which one is that? Is it primary, secondary, tertiary, or quaternary? Quickly, students, quickly in the chat. Tell me which protein is going to be, which structure of protein is going to be, right? Which protein structure is going to be bioactive in nature? Any of you, quickly. It is going to be tertiary structure. Is going to be bioactive in nature. Okay. In lipids, students, in lipids, we have fatty acids and glycerol, right? Glycerol, students, glycerol. What is it? What is the other name of glycerol, students? What is the other name of glycerol? Quick, students, quick. What is the other name of glycerol? Glycerol is also called as what? Glycerol is also called as what? Trihydroxypropane. Now, students, what will happen if I take a fatty acid and if I mix it with glycerol? If I take a fatty acid and I mix it with glycerol, what do I get? Quick. Fatty acid plus glycerol. I am adding both of them. I will get glyceride. Monoglyceride, diglyceride, triglycerides. Okay, please remember all this. Yesterday only you learned all this. Okay. Okay, see this question, students. Answer this question, all of you. Answer these questions. Oh, I didn't tell you, students. See, first statement makes sense. Okay. I'll not tell you anything. You start. Do it on your own. First statement is correct. But students, in second statement, this is the clue here. Just read this and tell me. Students, this is correct. Okay. But students, do you think cis phase and trans phase, are they identical? No, they were not identical. Cis phase and trans phase, they were actually different from each other. No. Cis phase is different, trans phase is different. They are not identical to each other. They are very different from each other. But they are interconnected. Students. This question. What is the important site of formation of glycoprotein and glycolipid in eukaryotic cell? 10 seconds, 10 seconds. 10 seconds, quickly student, 10 seconds. What is the most important site of formation? The Golgi bodies here. Friends, can anyone tell me in polysomes, what do we have? Single ribosome or many ribosomes? One ribosome or many ribosomes? Okay. Now, so two members of the gang done. Endoplasmic reticulum done, as well as the entire endoplasmic reticulum, as well as Golgi complex done. Next is called as lysozymes. Lysosome is called a suicide student, suicidal bag. Sad life it has. Suicidal bag has a very sad life. Okay. Suicidal bags and lysozymes are formed by the budding of vesicle from the Golgi body. From the Golgi body, they will bud off and they'll be produced. Okay. They have something called as lysosomal vesicles has something called as hydrolytic enzymes. Example, hydrolase, lipase, protease, carbohydrates. All of these enzymes are present inside the lysosomes and all of them will work in acidic medium. If you change the pH, they will not work. They will not work. Okay. Now I will show you how they bud. I'll show you how do they bud here. See, students, you can see here, this is the entire nucleus here. This entire thing is the ER here. From ER, let's say we get a protein here. Protein. Okay. Now protein is entering in the form of vesicle here. Now students, can anyone tell me which phase is this? This right here, is it cis phase or trans phase? Quick students, is this cis phase or trans phase? This right here is the cis phase. It is entering through cis phase. Nicely packaging is done, and from the trans phase it is coming out. It is coming out here, and this small vesicle is called as the lysosome. Called as the lysosome right now. Cis phase and trans phase. Okay, that is how protein packaging happens. That is how protein packaging, entire packaging happens here. Okay, entire packaging happens. Not cis. Okay. Question, all of you. Question on the board. If the pH in the lysosome is increased to alkaline, what will happen? Hydrolytic enzymes will function more efficiently. Hydrolytic enzymes will become inactive. Lysosomal enzymes will release into cytoplasm. Lysosomal enzymes will become more active. Friends, very easy, right? Very easy. Yes. Question number 2. They become inactive. Hydrolytic enzymes becomes inactive. The pHs become alkaline because they only can work in acidic pH. Which of the following statement is not correct? Statement is not correct here. Lysosomes are formed by packaging of endoplasmic reticulum. Lysosomes have numerous hydrolytic enzymes. Yes, correct. Lysosome enzymes or hydrolytic enzymes, lysosomes are active during acidic pH. Correct. Lysosomes are membrane bound structures. Yes, they have membrane bound structures. But are they a result of packaging of endoplasmic reticulum? No. They are a result of Golgi complex, not endoplasmic reticulum. So option number 1. Option number 1 is the answer. 1 is the answer. Okay? Okay. Next is called as vacuole. Students, vacuole is the large dustbin here. Vacuole is the large dustbin here. Whatever is not required, it is dumped in this dustbin. In the case of plant cell, and especially in the case of plant cell, they have a big dustbin, very big dustbin. Okay. So membrane bound shape, membrane bound structure, a membrane bound space of the cytoplasm is there. Here there is a single membrane called as tonoplast. Can all of you see? And around the vacuole, the layer is called as tonoplast here. Okay. It has excretory product. It has water sap. All the waste is dumped here. All the waste is dumped here, sir. Is the toilet only. Okay. In plants, 90% of vacuole of 90% of the volume of the cell is only vacuole. Can you see here? It's like, you know, one round big head for a small body. Okay. So for this entire cell, 90% is occupied by the vacuole only here. That is what is happening. Okay. Vacuole itself. Okay, students. Okay. Now what happens here, students? Because of the vacuole, right? All the inside the vacuole there will be high concentration of ions. High concentration of ions can be there inside the vacuole, and it always happens from the low concentration to high concentration they're going. Reverse they can go. That is because of vacuole. That is because of vacuole. In the case of plant cell, it produces a lot of waste. In the case of animal cell, it is more efficient. Okay, animal cell is more efficient. Okay. No, there are different types of vacuoles here. Okay, there is something called as your sap vacuole in the case of plant cell. In the case of amoeba, there is something called as contractile vacuole, and in the case of your again amoeba as well as your paramecium, we can see something called as food vacuole here. Okay, we can see something called as food vacuole here in the case of your amoeba here. Now, students, understand here that in the case of your vacuole here, amoeba, what is the main function here? Contractile vacuole has two main functions. Okay, it can undergo something called as osmoregulation, help in osmoregulation as well as excretion, as well as helps in excretion, as well as excretion. That is the main function of contractile vacuole, osmoregulation and excretion. Okay. Now we come to one structure called as mitochondria now. Mitochondria. Okay. Can I ask all of you a simple question here, students? In which cell do you think you can find more mitochondria? A dividing cell or a mature cell? Students, tell me, students, in the chat, quickly, students, where do you find more number of mitochondria? Let's take a germinating cell, like during seed germination. During seed germination, right, when seed is germinating, do you think that has more mitochondria or a normal mature, let's say a leaf cell? Let's take a parenchyma or any type of normal cell that will have more number of mitochondria. Always remember, students, the number of mitochondria will vary. Number of mitochondria will always vary, and number of mitochondria will be high wherever demand is high. Demand of mitochondria is high. For example, demand of energy is there. In the case of germinating cell, it has more energy demand, so more mitochondria can be there. Okay, one small concept here for all of you. Okay, it is also called as power of the cell, students. Why? Because it can produce ATP. It can produce ATP in electron transport chain where oxidative phosphorylation happens. Now, can anyone tell me which is the final electron acceptor in the case of ETC? Who is the final electron acceptor in the case of ETC? Quick. Christina, don't know. Let's see. Students, as you can see today, there's no energy with me because I'm a little sick. So there's no energy. Oxygen. Exactly. Oxygen is the final electron acceptor in the case of ETC. We say, right, aerobic respiration. We say aerobic respiration. Why? Because oxygen is involved there. Oxygen is involved there. Okay. Side for aerobic respiration. Divides by fission, double membrane structure. Sausage say diameter they will not ask you. Don't worry, diameter they will not ask you here. Students, can anyone tell me this link reaction happens inside mitochondria? True or false? Link reaction happens inside mitochondria. True or false? Quickly. Quick. As you can see, students, here we have the entire mitochondria. It has double membrane. We have outer membrane as well as inner membrane here. Outer membrane and inner membrane is present here. Between the two membranes, that is called the intermembrane space. Then there is entire matrix here. In this matrix, all the Krebs cycle, link reaction, all of it happens here. All of it happens here. Okay. And here there is protein foldings here. There are something called as protein foldings right here. Protein foldings right here. Yes. These protein foldings are called as crista. Okay. And this crista is different from the cisternae. Cisternae was where? In the case of Golgi complex. Here we have crista here. Okay. We have crista here. Okay. This is the diagram again which I have drawn for all of you. You can see outer membrane, inner membrane. We can see here double-stranded DNA with no histone protein. We can see ribosomes here, that is 70 ribosomes can be seen here. Then there is a lot of matrix here, Then what is the function of crista? Increase the surface area. It can increase the surface area here. Okay. Outer membrane is more permeable because of porins present here. And here we can see multiple racket-like structure called as your ATP synthase. Small structure called as ATP synthase, right? ATP synthase is also called as F0, F1 particles or oxysomes or elementary particles. These are different names of your ATP synthase. Now students, do you study about ATP synthase in your photosynthesis chapter? Tell me yes or no, all of you. Do we study about how ATP is formed? Right? I've taught you, right? ATP is formed here with the help of your, in the case of your chemiosmotic hypothesis. In the case of chemiosmotic hypothesis, we learn there how ATP is formed. We also learn it in where? In the ETC. In ETC also we learn how ATP is formed. That is because of proton gradient. Whenever there is a formation of proton gradient, right? Whenever there is transfer of 4H plus, 1 ATP is formed. 4H plus and 1 ATP is formed. Remember that. Now students, next thing we come to plastid. Next thing we come to the plastid, students. Now again students, this plastid right here, is it found in only in plant cell? Is the plastid found only in plant cell, students? Tell me yes or no. Only in plant cell? Plant cell and certain euglenoids, and also in the case of euglenoid, students. Here we have something called as some pigments can be present here. Certain pigments can be present here, students. These pigments can give certain color here. Pigments can give certain color here. Okay, pigments can produce certain color. Okay, certain color. For example, students, what are the plastids here? First, initially we learn about something called as chloroplast. All of you know chloroplast, the green pigment, right? Chlorophyll A, universal pigment, right? Chlorophyll A can give a green color, helps in your trapping of light. Okay. Then chromoplast, carotenoids, xanthophyll, two types are there, right? Chromoplast can give yellow, red, orange color. It can give. Then we have something called as leucoplast. What does leucoplast do? Leucoplast are three types: amyloplast, aleoplast, aileronplast. Now students, all of you know, right? Amylopectin. All of you know what is amylopectin? Amylopectin is part of your starch, right? A type of starch is there. Aleo, oil. It has oil storage, starch storage. Aileron layer. All of you remember in the seed, when I was teaching you monocot seed, right? Aileron layer can store proteins. It can store proteins. So all of these are different plastids which can store different types of substances. Okay. Amylo is going to be starch. Aleo is going to be oil and fat. Aileron is going to be proteins. Aileron is going to be proteins here. Okay. Leucoplast. Leucoplast will not give you any color. Remember, leucoplast will not give you any color here. Okay. This is plastid here. Now students, can anyone tell me right here, where does the dark reaction takes place? Anyone? Does a dark reaction happen? Does the dark reaction takes place in the here granum, thylakoid or stroma? Where does the dark reaction takes place? You tell me, sir. Dark reaction happens in the stroma right here. In the stroma, all the dark reaction takes place in the stroma. Okay, dark reaction takes place in the case of stroma right here. Okay, light reaction, students. Do you think light reaction, students? Do you think light reaction happens in stroma lamellae? Light reaction happens in stroma lamellae. If yes, tell me, in stroma lamellae, do we get cyclic or non-cyclic? Which one happens here? Tell me. In the case of stroma lamellae, do we get cyclic or non-cyclic? In the case of stroma lamellae, we only get cyclic. Why? Because only PS1 is present here. Only PS1 is present in the case of stroma lamellae. So only cyclic can happen here. Okay, cyclic can happen here. Okay. Answer I want quickly. over include this, no? Students, remember the same type of thing is present in the case of mitochondria also. I told you here, right? See, in the case of mitochondria also, double-strand DNA is present. Double-stranded circular DNA. Double-stranded circular DNA. Okay. Solve this question. Take your time. Take your time and answer this question, students. Take your time. Done? Done, students? See, chromoplast. Carotenes. Amylo is going to be starch. Aleo, oil. Aileron is going to be protein. Okay. A is going to be 4. Correct. B is going to be what? Amylo is going to be starch. B is going to be 3. Okay. C is going to be what? Aleo is going to be oil. C is going to be 2. C is going to be 2. Aileron is going to be protein. Aileron protein. This is 1, that's for sure. D is supposed to be 1 only. Done, students? How many of you got it wrong? Students, very easy, students, very easy this is. See, even if you do not know this much, you can tell this much, right? I told you, amylopectin is going to be starch. Aleo, oil, oil. Protein is going to be this one. Chromoplast is going to be carotenes, color pigment. Okay, I think some of you got it wrong. Okay, now we come to ribosome, students. Ribosome, right? In ribosome, students, you need to know one thing here. If they can ask you by chance, who discovered ribosome? George Paldane discovered the ribosome. Or they can ask you what a ribosome has. What is present inside the ribosome? Okay, what is present inside the ribosome? They can ask you. What is ribosome made up of? Ribosome is made up of amino acid plus RNA. So RNA plus amino acid makes up the entire ribosome. Okay, makes up the entire ribosome. Or they can ask you. Or they can ask you this structure right here. This structure right here. What is the, ribosome in the case of your eukaryote and prokaryote? You should know 70S is present here, 30 and 50. 80 is present here, 60 and 40, 80S. So 80S in the case of eukaryotes, 70S is in the case of prokaryote. Okay, in the case of prokaryote, students. Now we come to something called a cytoskeleton. Now we come to something called, students, cytoskeleton. What is cytoskeleton, students? Cytoskeleton is nothing but it is a network of protein basically. It is a network or framework, okay, made up of protein. Here there are mainly 3 types are there. There is something called as microtubules, microfilament, intermediate filaments. Okay, intermediate filaments. Okay, there is microtubules, microfilaments, and intermediate filaments. All 3 can be present. 3 types are present under cytoskeleton. Now, what they can ask you here is a function. They can ask you function, or sometimes they can ask you the function of cytoskeleton. Very simple again, students. Very simple functions. Okay, the main function is mechanical support. Mechanical support can be there. Yes, mechanical support can be here. Apart from the, students, it can also help in motility. Apart from the, students, it can also give cell shape. It can give a cell shape, can be given here. Cell shape can be given here. Okay, so cell shape, motility, and mechanical support are the three main functions of cytoskeleton. Okay, there are three main types here: microtubules, microfilaments, and intermediate filaments here. Now here this is very important because students, when you learn about the structure of cilia and flagella, right, the structure of cilia and flagella mainly comes from here. So please learn this at any cost. Okay, see, there is something called as microtubules are mostly hollow, microfilaments are solid, while intermediate filaments are going to be hollow again. Unbranched, also unbranched, and unbranched. These are sometimes they are branched also. They are non-contractile, contractile, non-contractile. This you don't have to remember. All you have to remember here is that, students, microtubules make up the cilia, flagella, and centriole. Cilia, flagella, and centriole are made up of microtubules. They are long cylindrical proteins which can come together to give a shape to something. Okay, they can come together to give a shape. Okay, give to a motility means movement, mobility. It can give movement also. Okay, movement. No, students. See, cilia and flagella we see now. What is cilia and flagella? Cilia and flagella is a hair-like outgrowth on the cell membrane. So on top of cell membrane, we can see cilia and flagella. Now let me show you a small image. You will get a better understanding. See this one, all of you. See this one, all of you. See right here. See, if we can notice here, students, if we can notice here, the cilia is very short, very short. The cilia is present throughout the body, Here we have a long flagella is present here. Long flagella at the end it is present. Now how can it move? How can it move, students? Has any one of you ever traveled in boat? If you traveled in any type of your boat like this, now what will happen, students, if I take both of them and I push it behind? I will go front, I will go front. That is called as oar-like movement. That is called as what? Oar-like movement. If I take it, hold it and push it back, the entire boat will go front. That is called as oar-like movement here. Okay, oar-like movement. So that can be observed in the case of cilia. Oar-like movement, shorter and smaller, longer, throughout the body, present at the end. So flagella is present at the end, cilia is present throughout the body. Cilia is present throughout the body, cilia is present. Okay, okay, students. Body. Now, understand the structure of cilia and flagella. Now, students, if I show you this diagram, and tell you this is cilia and flagella, do you understand, students? Can you understand anything from this? Like, from which angle does it look like a cilia flagella? From which angle does it look like cilia and flagella? Tell me in the chat. Any angle it looks like? No. No, so I will show you a small animation for this. You will get a better understanding, students. All of you, look at this. Students, see this. This right here is the flagella or cilia flagella. And you can notice here, students, this bottom part is called as basal body. It is called as basal body. Okay. Now from this basal body, this entire flagella arises. Okay, flagella arises. Now, students, in this flagella, understand that on this flagella, if you see the internal structure, can all of you see here? This part, you see this part here. If you see from the top view, the internal structure, we can see arrangement of small tube-like structure, which are hollow in nature. These small tube-like structures are called as microtubules. So in cilia and flagella, microtubules are arranged together here. They are arranged together. So if you can see this diagram right now, this is the magnification. This is the magnification of this top view right now. Now, if you see this image right now, you tell me, students, does this look like your diagram from NCERT? Like, yes, sir. This looks like your diagram from NCERT right now. Okay, diagram from your NCERT. So, if you want to understand cilia and flagella, you need to get the perspective. How is cilia and flagella looking like? Okay. Now, students, see this. Now, let's go back now. Now, does this make sense now, students? Tell me in the chat. Tell me yes or no. Does this make sense now, all of you? Yes. Does this make sense now? See, this right here is a top view. This right here, students, is a top view of the cilia and flagella. Top view of cilia and flagella right now. Okay. Now, if you can notice here, students, this entire thing is made up of what? This entire thing is made up of cilia. Entire thing is made up of microtubules. Okay. You can see here. You can see here, all of you. Microtubules everywhere. Now, let's count the microtubules present here. Okay. Let's go to my diagram. If you see my diagram, you can see here, students, 1, 2, 3, 4, 5, 6, 7, 8, 9. There are in total, right? In total, 9 microtubules on the periphery, and all of them are present as doublet here. All of them are present as doublet here. Okay. So there are 9 periphery. So 9 periphery, 9. Now, if you see in the center again, students, there are 2 microtubules in the center. In the center. Okay. Now, these 2 are called as central microtubules. The outer part is called as what? Peripheral microtubules. So 9 plus 2 arrangement. 9 plus 2 arrangement. Now, these 2 microtubules are joined together by a small bridge here. Bridge here. Okay. Now, students, understand. Okay, understand. Here, these microtubules are joined to the central sheath by something called as radial spoke. Can you see, student, this one? This part here. This part here is called as what? Radial spoke. It's called as what? Radial spoke right here. Individual called as radial spoke. Now, between first and second, between the 2 different peripheral microtubules, in the middle there is orange color part. Can you see orange color part? That is called as a linker. That is called as a linker. Okay. And this entire thing is called as what? Entire thing is called as what, students? Core or axoneme. It is called as what? Core or axoneme. Core or axoneme. The entire part. So this is the structure of cilia and flagella. Cilia and flagella. Okay. 9 plus 2 arrangement. 9 plus 2 arrangement. Okay. So this is how they can ask you question here. Who can tell me the answer, students? Let's see. Which of the following functions is carried out by cytoskeleton in a cell? Function: transportation, protein synthesis, nuclear division, motility. See, I told you it can provide mechanical strength. Yes, it can provide mechanical strength, or it can give motility or cell shape. Okay. Okay, students. So motility is the answer here. Motility is the answer. Now, what about centrosome, students? What about centrosome? How is centrosome here? Okay, understand, all of you, understand. How exactly centrosome? Here, centrosome is also made up of what? Microtubules. Entire thing. Okay. Now, if you look at a centrosome right now, this entire thing, if this is a cell, this entire thing is called a centrosome. The entire structure is called a centrosome. Now, here, understand, students, one centrosome is made up of 2 centrioles. One centrosome, this entire thing is called a centrosome. This entire centrosome is made up of 2 perpendicular structures called as centrioles. Can all of you centrioles? Yes, this is called centrioles here. Now, this entire centrioles in a cartwheel-like structure which are perpendicular to each other. And here in the centrosome, there's an outer membrane-like structure. There's no membrane, but membrane-like structure called as amorphous pericentriolar space, or called as kinoplasm, or also called as centrosphere here. Centrosphere. Okay. It can also be called centrosphere here. Okay. Now, this is a centrosome. Friends, can all of you see here? See here. Here we have 2 centrioles here. 2 centrioles which are perpendicular to each other. Perpendicular to each other right now. And if you focus on one of them, students, if you focus on one of them right now, see this. Can all of you see? They're hollow tubes. Hollow microtubules arranged everywhere. And you can see, students, here we have periphery are present, but central there is nothing here. Central nothing is there. In this periphery, if you count 1, 2, 3, 4, 5, 6, 7, 8, 9. Again, in the periphery, there are 9 here, but the difference here is that, students, all of these are present in triplets. All of these are present in triplets here. Triplets 1, 2, 3, 1, 2, 3, 1, 2, 3. Okay. So that is the difference between your cilia and flagella, as well as compared to centrioles. So in the case of centrioles, here we see 9 periphery present as triplets, but in the center we don't see anything. It is 0 in the center, 0 center. Now again, students, here the middle part is called as hub. This middle region is called as the hub here. Hub. Now this periphery microtubules are connected to the center by radial spoke. Again, radial spoke is present here. 9 plus 0 in the case of centriole here. Students, is this centrioles present in the case of plant cell? Tell me yes or no. Are they present in the case of plant cell, students? Tell me, students, are they present in the case of plant cell? No. There are 3 tubes, yes. 9 are present outside, but as a triplet they are present, as a triplet. That's why 3 tubes. Students, remember, students, if in this structure of the entire centriole here, this is called as a basal body, right? This here basal body is actually, this basal body here actually is 9 plus 0 arrangement, and the top part is 9 plus 2 arrangement. Okay, so basal body is similar to centrosome, or sorry, centriole. Top part is flagella, normal flagella or cilia. 9 plus 0, 9 plus 2 arrangement can be seen here. Okay, students. See, this is how it helps in cell division. This is how it helps in cell division right here. You can see from one centriole, from this is 2 centrioles here, entire thing is called a centrosome. We can see development of, we can see the formation of spindle fibers here. Spindle fibers are released from here. Spindle fibers are released from here, from the entire centrosome. From the entire centrosome, spindle fibers are released here. Okay, released. Now, students, now we come to the last part called as nucleus here. Nucleus, who can tell me, students, who discovered nucleus? It was discovered by Robert Brown. Robert Brown in the year 1831. So long ago, students, so long ago. 1831 by Robert Brown. Here, students, apart from this, there is something called as chromatin also. Chromatin here is the thread-like structure, and chromatin was discovered by Fleming. Was discovered by Fleming, students. Friends, give me a minute. Fleming, students, Fleming. Okay. He discovered the entire chromatin by dyeing something, by using a basic dye. Using a basic dye, students. Now, this is the structure of entire nucleus, students. Okay. And here, students, you can notice that, okay, here you can notice very clearly that in this entire nucleus, there are 2 layers here. Outer layer, inner layer. Outer membrane, inner membrane. Between the 2 membrane, the space is called as what? Perinuclear space, right in the middle. Okay. In this perinuclear space, there are small gaps here, small holes here. The small gaps is called as what, students? Small gaps is called as nuclear pore here. Nuclear pore. And this nuclear pore is important because it helps in transportation. It helps in transportation of your mRNA from inside to outside. Inside to outside. Okay. Okay, students. Now, next thing is called as nucleoplasm here. What is nucleoplasm? Nucleoplasm is the cytoplasm of the nucleus. Cytoplasm nucleus. Then we have something called as chromatin here. Chromatin. Now you can notice here, students, we have chromatin and not chromosomes. Why? Because chromosomes are present in dividing stage. In your prophase, metaphase, anaphase, we have chromosome. Since this is right now in interphase, we have chromatin here. What is the chromatin? Chromatin is nothing but it is a genetic material which has the DNA, histone proteins, non-histone proteins. Everything is present in the chromatin. In the chromatin, students. Okay. Then in this entire nucleus, you can see from the outer part, we can see the ER development also. ER development. Now here we have certain structures called as your nucleolus. What is the function, students? The function of nucleolus is they are the factory of rRNA. They'll produce rRNA eventually. Okay, they'll produce rRNA here, nucleolus. Okay, you can see in this entire nucleus, we can see nucleolus in the right in the middle, which can produce rRNA. We have chromatin everywhere inside. Okay, this I told you, right? Nucleus as in the red blood cells, as well as the tracheids, and tracheids we do not have any type of your nucleus here. Gone. See, student, this is the chromatin. You can see this is the chromatin right here. If this is pulled, this is chromatin right now. Chromatin. Chromatin has your genetic material. Okay. The slide is missing, students. Okay. You can see here, students. There is something called as heterochromatin, euchromatin. What is heterochromatin? Heterochromatin is tightly linked. Heterochromatin is very tightly linked and it is inactive in nature. Inactive. This is going to be inactive in nature. While euchromatin is very lightly stained and it is active in nature. It is active in nature. It is active in nature. Now, if you look at the chromosome here, students, chromosome. A chromosome has something called as a primary, yes, Maggie only. A chromosome has something called as a primary construction right in the middle. Primary construction is called a centrosome. It's called a centromere, right? Called a centromere, and is inactive in nature. Why? Because it is made up of heterochromatin in general. Okay. Around the primary construction, there is something called a protein cup-like structure called as the kinetochore. Right. All the spindle fibers will always attach to kinetochore. Remember, always to kinetochore. It will never attach to the centromere. Never. Now these are called as sister chromatids, which are present together. Sister chromatid. Then there is something called a secondary construction on top. Secondary construction. That is called as a satellite here. Satellite. Now these ends of a chromosome is called as telomeres. Telomeres are called as the ends here. Okay, or end here. And remember, students, this entire thing in the middle is your euchromatin, which is transcriptionally active here. Euchromatin. Somewhere in the ends we have heterochromatin like this. Heterochromatin in the end somewhere here. Okay. See this. There is something called, this is called as P arm, this is called as Q arm. If they ask you by mistake also, P arm is on the top, Q arm is at the bottom. P and Q. P and Q. There are two present here, no? Both sides. Okay. Now, based on that, we have multiple types of chromosomes. Based on the shape of, we have different shapes of chromosome. Now, students, can anyone tell me in which stage of mitosis can I study all this? In which stage of mitosis can I study all this? Is it metaphase or anaphase? See, I always tell students, in metaphase we can study shape of, in metaphase we can study morphology, karyotyping in metaphase, but this has to be done in anaphase. This has to be done in anaphase, students, anaphase. This has to be done in anaphase right now. Metacentric, submetacentric, telocentric, acrocentric. In anaphase. You can see here also how it is done. Telocentric, acrocentric, submetacentric, L shape, V shape, I shape, J shape, chemistry. Don't tell metacentric. Metaphase we can study what? Metaphase we can study the morphology. Shape can be studied in, shape can be studied where? In anaphase. Remember that. Okay. Okay, students. In anaphase we can study. Let me answer this question. Let me answer, students. Here we can see that they're asking the question is asking you, which of the following represents sister chromatids here? See, this is sister chromatids. This is non-sister. One from here is non-sister. Middle part is called a centromere. There again one chromatid only. But look at B here. B, one this one, this one. They are called as sister chromatids, right? They are called as what? Sister chromatids right here. They are called as sister chromatids right here. So what is the answer? What is the answer, students? Option number B. Okay, option number B or 2. Look at this question, students. Cilia and flagella, what do we see? In cilia and flagella, we see axoneme, or the core. Endoplasmic reticulum, what do we see? We see ribosomes. Mitochondria, cristae. Kinetochore, spindle fiber. Okay. What is the answer? Option number 2. Then there is something called as microbodies in the endocytosis. What is microbodies? There are small vesicles called as microbodies that contain your enzymes. They're present in plant and animal cell. That's all. Small structures which has vesicles, which are present in plant cell and animal cell both. Oh, what is this? So much of wind from the AC. Okay, plant cell and animal cell both. Solve this question, students. Solve this question. Quick question. Nucleolus, I told you, site for rRNA. Okay. Centriole is cartwheel. Okay. Leucoplast, storage. Golgi body, site for glycolipid. Golgi body, site for glycoprotein. So nucleolus is going to be your RNA. A is going to be 3. Okay, B is going to be 2. A is going to be 3, B is going to be 2, C is going to be 4, D is going to be 1. So option number 4. Student, this question right here is 2024 question. Just saying. Look at this question. Axoneme is going to be the cilia and flagella. Cartwheel is going to be centriole. So A is going to be 2, B is going to be 1, C is going to be 4, D is going to be 3. C is going to be 4 and D is going to be 3 here. So the answer is going to be very easy now. A is 2, B is 1 only, you got the answer. Option number 3. Three is the answer, you got it there only. Which of the following statement is incorrect statement? Incorrect statement, students. Incorrect. Okay. Perinuclear space forms a barrier between material present inside of the nucleus and that of the cytoplasm. Okay. Nuclear pore acts as a passage for protein, RNA in molecular both directions between nucleus and cytoplasm. Okay. Mature CO tube elements possess a conspicuous nucleus. No. I told you, students. Mature CO tubes do not have nucleus. They do not have nucleus at all. Lucky Chan, yes, you can. You have to do more question practice. Okay. You should do more question practice. It won't be easy. Do more question practice like this. The biosynthesis of ribosomal rRNA, that is rRNA, ribosomal, that is rRNA is made where? Factory of rRNA, that is going to be nucleolus. Students, nucleolus. Last question. Students, I don't think so I can take cell cycle. My fever is coming up. So let's see. Which of the following pair of organelles does not contain DNA? See, mitochondria has DNA, gone. Mitochondria has DNA, gone. Chloroplast has DNA, gone. Lysosome and vacuole do not have DNA. They do not have DNA. 4 is the answer. Okay, 4 is the answer, students. Yes, students. Very fast, I know. Students, I have done all this. All of you know, right? If you want to check in detail, students, if you want to check this in detail, just type cell cycle and cell division, right? Cell cycle and cell division backlog series. There I've taken the entire chapter for 3.5 hours, 3 hours, I guess, 3 hours, entire chapter 3 hours. So please check that once, okay? Because I think now the reason I went a little fast in the end was because I think I'm getting fever now. So I don't think I can take cell chapter. I'll just call Gopika ma'am and I'll just go sit down for some time, okay? So thank you so much, students, for today's class, right? I'll meet you in the next class, students. Until then, take care, all of you, right? I'll just call Gopika ma'am and I'll come back, okay? Students, ma'am is like give you a lunch break, okay? Ma'am is like give a ma'am also told us like give him a lunch break. So we'll give you lunch break for 30 minutes. Go eat and come, students. Class will resume back. Okay. Okay. So class will resume back at 2 p.m. So I will see all of you at 2 p.m. right? Okay. So I'll see all of you at 2 p.m. So Gopika will come at 2 p.m. Okay. Notes will be given in Telegram channel. Don't worry. Notes will be given to all of you. You have to chill little. Okay. So I'll come back later, students.
ಬಸವರಾಜ್ ಸರ್ ಬ್ರೋ ಇದು ಸ್ಟಾಪ್ ಸ್ಟ್ರಿಂಗ್ ಮಾಡ್ಬೇಕಾ ಬೇಡವಾ ಇಲ್ಲಿ ಇಲ್ಲ ಸ್ಟಾಪ್ ಸ್ಟ್ರಿಂಗ್ ಇದೆ ಅದಕ್ಕೋಸ್ಕರ ಆನ್ ಇದೆಯಾ
Let me just download the PPT. One second, students. Can you all see me clearly? Hello, hello. So sorry, it became a little late. Okay, because we had just gone for lunch. So are you all ready? All of you ready? One second, I'll just share the link on Telegram as well. Okay. Hello, hello. Okay, so students, we will start with cockroach and frog first because animal tissues, we just did it very recently, right? I think a week back we did the animal tissues and most of the students told me, ma'am, they do not have any doubts with animal tissues, but many of them have problem with cockroach and frog. And also, according to NDA, cockroach and frog questions comes more repetitively compared to your animal tissues, okay? Animal tissues lays the foundation, students, but still, you know, it's important for you to know the topic. So I have put my notes. Don't worry, I have put my notes. Okay, shall we start? And last class, frog got, frog jumped out of my live class and the stream had ended. So that's why we will start with cockroach and frog. Okay, so keeping that in mind, I have put my handwritten notes here so that you can have all of it here. Everything is written. Okay, everything is written. This is the best notes that you can have. All you have to do is just revise it. Okay, because most of you attended my last class, which happened a week ago. So we will first start with our cockroach. Okay, or do you want to start with frog? Okay, we can do it both the ways, no problem. It's all individual topics. Okay, it's all individual topics. Okay, so let's do one thing. Okay, let's start with cockroach. Okay, because cockroach is going to be very fast and easy for us. So we will start with cockroach. Ready, all of you? Yes or no? Can I see some energy in the class? Yes, come on, some energy in the class. Okay. Yes, where is frog and cockroach? Okay, they were there in my last class. Okay, I think you people didn't like the video or share the video with your friends that I did not get enough famous for them to join. Okay, so today we will finish frog at any cost. Okay, chalo, let's start. So students, what is cockroach? The kingdom. Kingdom is obviously going to be Animalia. Right, kingdom is going to be Animalia. What is the phylum? Arthropoda, the largest phylum. And what makes Arthropoda the largest phylum? The presence of chitinous exoskeleton, right? Yes, all of you have to continuously answer. Recently only you studied cockroach with me. Now class is going to be students Insecta. And again, one of the reasons why Arthropoda is the largest phylum is because of the class Insecta. Because you know 2/3 of the organisms is insects, okay? Yes, next is scientific name. I told you it sounds like a Starbucks drink, but what is the name? Periplaneta, okay, Americana. Periplaneta Americana. This is the scientific name of cockroach. Now, adding on to this, students, you should always understand that Arthropoda is one of the largest phylum because of two reasons. Because of their chitinous exoskeleton. Yes or no? Chitinous exoskeleton plus what? Plus what? All of you tell me. Arthropoda. What is the terminology? Arthro, jointed. Poda. Okay, not like go, not that Tamil poda. Legs. Okay, jointed appendages or jointed legs. This is the two. This is just the separation of the names. Okay, just the separation of the name. So we understood that this name is Periplaneta Americana. Now, what do you know about the general characteristics? All of you tell me in the chat box. What do you understand about the general characteristics of cockroach? See, students, first of all, understand cockroach is usually which color do you see? Ma'am, I see brown. Okay, I have also seen black bodied, right? Brown or black bodied cockroach. These people, mainly these cockroaches, are nocturnal animals. What is nocturnal, students? Like you people only, cockroaches also are nocturnal. That means what? That means what? That these people also night only they become active. You people also, no, full day you will be like, why to study, what to do. Suddenly when 10 o'clock live is there, means everyone will be in that live. Why? Because you people also are nocturnal animals. You're active during the night. So this nocturnal means they're active during the night, and that's why you spot them in the kitchen when you're going to steal food. So brown or black bodied, and they're nocturnal animals. And students, these people are omnivorous. Okay, these people are omnivores, but more than omnivores, they can also be cannibals. That means what? They can feed on their same species. Now, do you know that cockroaches are present in other colors also? You will be like, ma'am, what are you saying, ma'am? Yes, they are present in bright yellow, red, or green colors. But not here. Okay, luckily it is not here. Because here if it was there, then people would have taken it and kept it as a museum piece. It is seen in the tropical regions. Okay, it is seen in the tropical regions. Okay, this board is not that smooth, so my handwriting is going to be a little, you know, a little worse than, you know, other times you don't know how I write. So it's going to be a little hard for me to write. Fine? Yes? Okay. So bright yellow. Very good. Yes, ma'am. Green color. Okay. So these people will be in bright yellow, red or green in color. Then what else do we know about their size? These people will range from 1/4 inch to 3 inches. Okay. These are all general thing. But see, a statement based question can come on this because they will be thinking that, okay, which student knows about this? So they might give based on that. And then your Periplaneta Americana is 34 to 53 mm in length with wings. Got it? With wings. Fine. Handwriting is legible. Okay. Fine. Mm in length or long. Okay, we can write long also. Long with wings. Done, done, done. All of you tell me in the chat box. Done, done, done. Now, one of the most, we can't say it's general characteristics, but we can say it is one of the prominent characteristics that we saw with Arthropoda. Okay, with Arthropoda was that their body division. Their body is divided into, students. What is that divisibility? Okay, head, thorax, and abdomen. Head, thorax, and abdomen. Is this possible to remember? Yes or no possible? Oh, this mic. Yes, tell me. Yes. Okay. Head, thorax and abdomen. Very good. Head, thorax and abdomen. Okay. So this is the, let's say, a general characteristics of cockroach. Okay. But are they going to ask you only general characteristics? No. They are going to focus on the specialized characteristics. So all of you tell me, what is the specialized characteristics of cockroach? Let's try with different pen students. It might work. Okay. So we understood that the body is divided into head, thorax and abdomen. But the most important thing that I told you when I spoke about, oh, this pen is bad. Okay, 1 second. We are figuring it out, how to write neatly. Okay. When I told you about arthropoda, I told you the arthropoda phylum is the largest because of what? All of you tell me in the chat box. Because of what? Phylum arthropoda is so famous. Not so famous, the largest phylum. Why? Come on, quickly in the chats. Yes, quickly in the chats. Thank you. It is because of their chitinous exoskeleton, right? Chitinous exoskeleton. So students, when we talk about this, all of you should remember the cockroach body has something called as plates, okay, which are made up of chitin. Okay, these chitinous plates are present and these plates are called as sclerites. These plates are called as sclerites. And these sclerites are present dorsally, ventrally and laterally in the cockroach. All of you tell me, I've taught you this again. Dorsally. How will you remember dorsally? T you will remember. No, DT you will remember. Tergite. So the structure which is present, the plate or the sclerite that is present dorsally is called as the tergites. Okay, tergites. Very good, very good. Okay, and then we have ventrally. Ventrally, what is it called? Ma'am, it is called sternites. Okay, VS. So DT, VS. Okay, remember like this. DT, VS. Okay, dog Tommy went scuba diving. Okay, I don't know for S for what, students. Okay. Yes. Good afternoon. Good afternoon. Yes. So dorsally, tergites, ventrally, sternites, and obviously laterally, it's called pleurites. Okay, pleurites. Now students, imagine all these pieces are there. Dorsal is there, ventral is there, lateral is there. Now just imagine I keep different pieces of paper and I tell you, okay, you walk with a piece of paper. Now one you will hold here, one will fall from the top. One you will hold here, one will fall from the top. So to make sure that all these plates are, you know, articulated, kept together, we have an articulating membrane. What do we have? We have an articulating membrane. What is that membrane called? All of you tell me in the chat box. It's called the arthrodial membrane. Arthrodial. Oh. Friends, you can follow my last time's notes itself because this notes won't be neat because of the board. Okay. Where do you get that? Here, I guess. Yeah. Okay. Arthrodial membrane. Important. Important. Okay. Yes. Okay. Yes. So here what is going to happen? Here what is going to happen? Dog Tommy went SLP. Okay. So dog Tommy went swimming in Lucknow Palace. Very difficult to remember. Okay, dog Tommy, at least you remember. Students, if you remember dog Tommy, at least you will be able to write dorsal properly, and obviously if you write dorsal properly, automatically you will be able to remember ventral. Okay, at least remember one. Okay, dog Tommy went swimming to Lucknow Palace. Okay, understood? Else say some places there, that also you tell me. Fine? Clear? Articulating membrane, okay? Articulating membrane, which is called the arthrodial membrane. I've put a star here, so that means it is very, very important. Fine? Now, okay? The same thing I have just written it here. So remember, tergum is dorsal, sternum is ventral, and pleura is lateral. Okay? Now we will understand the cockroach structure. More than cockroach structure, we are going to understand. Okay? It is a difficult trick, ma'am. Then please give me a trick. That's why I told you it's difficult. Just remember dog Tommy. Tommy you will remember. That song was there, no? Very famous song. What is that? Sadda Kutta Tommy. Sadda Kutta Kutta. Something like that. One song was there. Remember all of you? So remember like that only. Dog Tommy. Okay. Now look here, students. The head region of the cockroach. The head region of the cockroach has compound eyes. What does it have? Compound eyes. Okay. It has antenna. It has mouth parts. Okay. These are the main things that are present in the head of the cockroach. So head of the cockroach is first of all triangular in structure, and these people have filiform antennae. They have compound eyes. Again, thorax region has classifications. Okay, that classifications we will come into. So right now, did you understand this much? Did you understand this much? Okay. Now students, head region. What and all head has? First of all, you should understand the head is made up of 6 embryonic segments. It's made up of fusion of 6 embryonic segments. Important, very, very important. 6 fusion of, okay, I'm adding it here, fusion of 6 embryonic segments. Okay, head is made like that. Then second most important thing, these people are triangular. The head is triangular and it is located 90 degree to the body. Okay, 90 degree perpendicular to the body axis. To the body axis. Look at the structure here, all of you. Quickly, look here. The head is triangular. Okay, it's looking at you. Can you see? Study, better study and like the video. Otherwise, it's going to come today itself. Okay, it's going to haunt you. Yes, so look here. It is at right angle to the body axis. Okay, it is right angle to the body axis. Now, students, when we move forward about the head, what do you have to understand? These people are made up of 6 segments, triangular, 90 degree to the body axis. Clear? What are the parts which are present in the head? What are the parts which are present in the head? Ma'am, you told us that head has, okay, head has, which and all parts? First, you start with the filiform antennae. Okay, what are these people going to do, students? Obviously, ma'am, they're sensory. Right, they're sensory in function. They will understand that, okay, is Madhumita standing in front of me or not? The thing will understand. Okay, the thing will understand. Clear? Yes. So, the filiform antenna will be sensory. They will understand that if something is happening in the front or there is some obstruction in the front, the antenna will recognize. Okay, they're sensory. Then second, you come, students, second. God, my handwriting looks terrible on this board. Yeah. Okay, so filiform antennae, sensory. Now, next thing that you have to understand is about their compound eyes. Okay, compound eyes. What do you know about their compound eyes? As I told you before also, their compound eyes, students, these people have low resolution. Low resolution. It does not have your iPhone, Samsung, your DSLR resolution. It has low resolution, but it has high sensitivity. It has what? High sensitivity. So, compound eyes have low resolution. Can you all see? Low resolution, high sensitivity. And then what does it have? It have 2000 ommatidias which are going to ultimately give that mosaic vision. So if this is the eyes of the cockroach, they have like this hexagonal structures. Okay, they have this hexagonal structures which are ommatidias, which will accumulate. Like in the sense, what will they do? They will get all the images and they will give a mosaic vision. Okay, mosaic vision. Have you all seen mosaic tiles? Okay, in olden days, it was there in our grandparents' house and all. Mosaic tiles we used to tell. Clear? Very good. Very good. Okay. Cockroach can be an enemy to boys also once it starts opening its wings. Okay. Don't be so happy also that, oh my God, I'm not scared of cockroach. Filiform antenna, compound eyes. Then what do they have, students? They have the mouth parts, which you have to understand with the help of an image. But understand the mouth is hypognathous mouth. Ma'am, what is this, ma'am? Such a big term you have told. Nothing, student. Hypognathous means the head is towards the floor. That means what? They are going to feed like this, right? It's not like this. It is bent like this and it is towards the floor. So, the consuming food will be easier for them. Consuming food will be easier for them. Clear? Okay? Yes. Now, look here, students. The mouth parts. The mouth parts of cockroach is important to understand, label and write it. See. First you have labrum. What is labrum? Ma'am, labrum is the upper lip. Labrum is the upper lip. Ma'am, what is mandible? These people are going to do the job of grinding. Okay, grinding. They are in a pair. Okay, they are in a pair. Then what about hypopharynx? It is going to act as the tongue. It is going to act as the tongue. Okay. Now, maxilla. Maxilla, students. Maxilla has maxillary pals. Okay. What do they have? Maxi. I'll write it outside. I think this will be fresh in my students' head because the crash course students had it just now and my YouTube students had it last week. Okay, this topic will be fresh in your head. Maxillary pals. And then you have labium, which is the lower lip. Okay, lower lip. So, a pair of maxilla, a pair of mandible, labrum, labium and hypopharynx. This is the entire thing. Clear to all of you? Yes or no? So, they have a pair of what and all? Come on, quickly tell me in the chat box. Starting with mandible, who is going to do the grinding. Mandible. So, like that. Okay, mandible. So, that is the thing. Okay. Yes. Then, students. Oh, I'm inside the mic is like, I will not let you stand properly. Okay. Yes. Mandible. And then they have pair of maxilla, which are sensory, okay, which are going to help with the taste and other things, senses, okay, clear to all of you. And they have labrum, labium and hypopharynx, okay, labrum, labium and hypopharynx. Did we miss something? Did we miss something? Think and tell me quickly. Yes. Ommatidia. Yes, 2000 ommatidias did we write here? Okay, we have to write here that these people have 2000 ommatidias. 2000 ommatidia. Okay, 2000 ommatidia. Done, done, done. Mouth parts is clear. Will you be able to identify labrum and labium? I told you. Labrum is just a pear-shaped structure. Labium is more complex structure. Okay, complex structure. So that complex structure will become the lower lip. The simple structure will become the upper lip. Then hypopharynx, tongue. Okay. Then you have mandible. So when you're stressing that word, mandible, grinding, maxilla, sensory, taste. Okay, all this will be done by that. Okay, clear to all of you? Yes, I just had my lunch and came. Okay, now students, did you all have your lunch? Now, thorax, very, very important thing about thorax is that thorax is divided into 3 segments, okay? What are they? This is the prothorax, this is the mesothorax, this is the metathorax. So, your entire thorax is divided into 3 portions. Now, students, remember, when even though I'm drawing like this, like a very sharp end, you should understand that above this, there is a head. Okay, above this, there is a head and this is the head, okay, with 2 eyes. Clear? So, this whole thing is divided into, first you start with pro, okay, you start as a pro and then you end up being a meta, okay, mesothorax and this is the metathorax. Now ma'am, what should I study more importantly from this? First of all, you should understand all these 3 parts, okay, all these 3 parts have a pair of jointed legs. This can you remember? A pair of jointed legs. Done? Okay. Next thing. Okay. 1 second, students. I forgot to get water. Okay. Chalo. Yes. So, a pair of jointed legs. Yes, a pair of jointed legs. Yes, ma'am. Then, students, other than that, here the most important thing is where are the wings present? Okay. First wing is going to be present in the mesothorax. First wing is going to be present where? In the mesothorax. Clear? First wing is going to be present in the mesothorax. Okay. Yes. Then second wing is going to be present in the metathorax. Second wing is going to be present where? Ma'am, it is going to be present in the metathorax. Clear? Now, which wing is going to do what? Okay, which wing is going to do what? So in this first wing, the orange color wing, so we will write one orange color dot. That first wing is going to be called, oh yeah, I bought my glasses, no, that's why I'm thinking why I can't see. Yes, it's a non-flight ring, flight, okay, flight wing. Yes, ask me that out, ask me that out. Yes, 300 likes target, quickly, quickly. Okay. Ah, very good. Tegumina. Okay, tegumina. Okay. So, mesothorax, the wing is going to be called as mesothoracic wing, and the wings on the metathorax is going to be called metathoracic wing. So, they'll have a pair of wings. Now, students, here you should understand this mesothoracic wing, no? This mesothoracic wing, which is present in the mesothorax, okay, mesothorax, is going to be called the forewings. Okay, it's going to be called the forewings, and this is also called as tegumina. Who? Not Jasmina, it's tegumina. Okay, this is called tegumina. And student, this tegumina is not going to do much job. Why? Ma'am, this is mainly for protection. This is mainly going to do the protection of your hind wings. Understood? This is going to do the protection of your hind wings, and these people, question will come on this. They are dark and opaque. Dark and opaque and leathery. Okay, these people are not for flight. Okay, they will not help in flight. Understood? They will not help in flight. Done, 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 done. Okay? Yes. Clear? Clear? Okay. Now moving forward, students. Moving forward about your metathoracic wing. Okay, metathoracic wing. I'm so sorry, I'm writing it everywhere. Don't worry. For notes, you can take my previous notes itself, because here writing is becoming very hard. Okay, writing is becoming very hard. So students, this metathoracic wing, what you need to remember, this wing is called as the hind wing. This wing is called as the hind wing, and this wing is going to be the one who is going to fly. That is the one that scares us, okay? But the wing is going to be transparent, okay? The wing is going to be transparent, and the wing is going to help us with flight. Can you remember this much? Yes, ma'am, we can remember this much, okay? It's not that difficult to remember this much. Done, done, done. Any doubts? Any doubts? Not join, exit. Okay. Yes, done, students. Clear? I hope no doubts is there. I have also, students, cockroach, I have also shared a handwritten notes, separate handwritten notes on my telegram. This is my telegram. You can join my telegram. I have given a handwritten notes of it, like in the sense I wrote it. This also I write with my hand only. It's not like I'm writing with any tool, but handwritten notes I have written. That means book and paper. Okay. That method I have written and given. Okay. Now, students, so thorax is clear. Any doubt in thorax? Prothorax, mesothorax, metathorax, all the three thorax regions have a pair of jointed appendages. Now, two of the thorax regions have wings, that is mesothorax have wings called as mesothoracic wing. Metathorax have a wing called as metathoracic wing. In that, mesothoracic wing is also called as the forewing or the tegumina, which is not used for flight. Okay, it is dark, opaque, leathery. They will ask you which is the wing which is dark, opaque and leathery. It is not used for flight. It is just going to do the job of protecting the hind wings. Clear? Yes or no? Clear? Okay. Yeah, if I don't wear glasses, then you will have to come here and take everyday classes. Okay. Eyesight will go. Understood? Okay. Now, students, here, look here. This is jointed appendages. This is one segment. Okay, this is another segment. Okay, and this is another segment. Okay, this is how jointed legs look like. Clear? Okay, fine, perfect, perfect. So this is your thorax for you. Now we will write about abdomen. Students, what do we know about abdomen? Here, abdomen, mainly you have to study few portions. Okay, first of all, you should understand your abdomen of cockroach contains, okay, 10 segments. 10 segments. This number you remember and keep 10 segments. Now, students, in abdomen, on the 10th segment, okay, on the 10th segment, you will see anal cirri. You will see what? You will see the anal cirri. Ma'am, is this anal cirri present both in male and female? Yes, it is present in male and female. And what are they going to do? They are going to help with capturing the vibrations. That is how they will get to know that, okay, Mintu is coming from far region with chappal. They'll get to know because of the vibrations. Clear? Yes or no? Half of the time I'm not in the screen, no, because I have to hold the board. Okay, yes, clear? Okay. Now, students, other than this, the most important thing that is present is about your brood pouch. So here, remember this number, 7th, 8th, 9th. Can you remember this three numbers, 7, 8, 9? Do I have to call a maths teacher for that? Do I have to call a maths teacher for that? No, ma'am, you don't have to call a maths teacher for that. Now, think of this 7th, okay, 7th sterna. First, I'll write here. All are sterna. Sterna means your sternum you studied, no? So, in that, your 7th one, okay, your 7th one is going to be boat shaped. Boat shaped. This much you can remember? Yes, ma'am, we can remember boat shaped. Now, they are saying that all these three segments, okay, 7th, 8th and the 9th segment together is going to form the brood pouch. Together going to form the brood pouch or the genital pouch. Genital pouch. Attaita, attama, attam, chestunda. I don't know, students. I keep learning the word, but I forget. Okay? Yes, clear? Clear? Yes. 7th, 8th, 9th. Okay? 7th, 8th, 9th sterna together forms the brood pouch or the genital pouch. And the 7th sterna, it is boat shaped. Okay? 7th sterna is boat shaped. Clear? Yes. Now, they are saying that anterior to this. They are saying that anterior to this. What is present, students? Come on, all of you answer me. You all know it. So, it is, they are saying that there is female gonophore. Gonophore means the opening. Okay, the opening. Female gonophore. Then they are saying there is spermathecal. Sperma. Okay, spermathecal. Okay, spermathecal. Again, spermathecal, you know, no? The spermatheca, which we study. So, spermathecal and your collateral gland. Okay, collateral gland. They're saying that these three things are present anterior to the genital pouch. Now, one more thing that you have to understand from this is your genital pouch is made up of two things. What are the two things they're made up of? Vestibulum and genital chamber. This is where the ootheca will be waiting, okay? Waiting for finding a place where they can lay the eggs, that is which has moisture content and where there is food content. Done, done, done. Done, done, done. Yes or no? Okay, we'll finish it fast, don't worry. Attaitu, ma'am. Okay, very nice. What do you tell for in Tamil? Tamil and Telugu, you tell me. Tamil and Telugu, you tell me. What do you tell for? How did you understand? Okay, did you understand? Now, students, when you come to males, males also have, yes ma'am, males also should have genital pouch. So, in male students, you have to remember some different types of number. You have to remember 9, 10. Okay, again you have to remember and 9. Now, since this 9 fellow is separate, you should understand that this 9 fellow is going to be made up of something else, right? So, this fellow and this fellow are separate people. What are they made up of? These two are made up of terga. Okay, and this fellow is made up of sterna. So, the 9th, 10th and the 9th segment in the males are always complicated, okay, is going to make their brood pouch or your, like a, okay, genital pouch we can say. Okay, genital pouch. Done, 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 done. Yes or no? Easy peasy lemon squeezy. Purinjada. Thank you, sir. Yes, Purinjida. Arthamainda. Similar to Kannada, no?
Which one, ma'am?
If you understood in Telugu.
Yeah. How are they doing, ma'am?
They're doing very excellent.
Very excellent, huh? Double, triple.
Double, triple, nothing like that. Something. They're very interested in cockroach. They love cockroach. They like talking about cockroach. They
Not
like
so
being
much.
scared about cockroach. I don't know. They're just very interested in cockroach. Whenever I teach cockroach, they're very excited. You see the chat box, for no reason they'll be excited.
Why there sparkles in the chat box for cockroach, ma'am?
They are so scared of cockroach that now they're trying to do black magic. Like in the chat box, they're like, cockroach is really nice. Cockroach is very nice. So that cockroach doesn't haunt them. Right? Okay? Yes. Students, sir is preparing few things for 30th March. Okay, so don't miss it. If you're in Bangalore, do come. Okay, yes. Now, students, genital pouch. Now, what is anterior to the genital pouch here? To the anterior portion, we have anus. Okay, anus, all of you know. Now, again, anterior to this pouch I'm talking about, we have again genital pore. We have gonopophysis. Okay, gonopophysis. These are the three things that are there. Okay. Yes, come on. Yes, okay. Clear to all of you? Now, I told you about anal cirri. Again, students, there is sexual dimorphism shown in cockroach. Okay, sexual. Now, tomorrow, if you hold, if you catch a cockroach, you have to check one thing. What is that? You will check if the males have, okay, if the males have something called as anal style. If the males have something called as anal style, which is not there in cockroach. Okay, and I have told you this before also. Males does style, okay, males does style. That is why they have anal style. Okay, again on the 10th segment, they will have this anal style structure and females will have only anal cirri. Is this clear? These numbers are very, very important. If you forget this, then gone. Okay, obsessing over cockroach will not work out. Now, going to the anatomy. Ready for the anatomy? Okay, yes. Yes, clear? Okay. Now, when we study about the anatomy or the digestive system, students, digestive system is very easy. Very easy, very easy. There's nothing to break your head about, okay? Even frog, even cockroach, nothing to worry about. Look here. The alimentary canal present in the body cavity is divided into three regions: foregut, midgut, and hindgut. Just remember this. Foregut, midgut, and hindgut. Okay. Now, we will learn about that by drawing it. Okay. I don't know how I'll draw it on this board, but yeah, let's try. Okay. Let's try. As I expected, students. As expected, not possible. Okay, I'll try my best. Okay, if some problem is there, you please forgive me. Okay. Did you understand the general characteristics and the specialized characteristics? If you have any doubts, ask me now. Ask me now. Yes. Yes. Look here, students. I have drawn a very fancy diagram for all of you. Now I'm going to explain it to all of you. Okay. Okay. Look here. I have drawn the entire digestive system of cockroach. Okay. Yes, entire digestive system of cockroach. Now this one, students, this is the esophagus. What is it? The esophagus. But before the esophagus, what will be there? The food would have entered through the mouth and it would have reached the esophagus through the pharynx. Any doubt in this? Any doubt in this? Anyone has? No, right? Esophagus. Okay. Yes. Clear? Yes. Quickly like the video, all conjuice. Okay, yes. Okay, now look here, mouth, esophagus, and this is the most important structure, the crop, which looks very similar to our stomach, right? A sac-like structure, okay? It is a sac-like structure. Now, students, this crop has a function. The crop function is storage. Okay, storage. Then from crop, when you go down, you will see one triangular structure which is called the gizzard. Now, this gizzard is also called as proventriculus. Proventriculus, which is going to do the job of grinding, and this you have to remember one very important thing. This proventriculus or your gizzard has 6 chitinous plates which can be called, or which will act like the teeth. Which will act like the teeth. Understood? Which will act like the teeth. So what will it do? It will do grinding. Okay. Now comes the main part. So students, by this we have completed the foregut. So this is going to be the foregut. So between the foregut and the midgut, the blue color that you're seeing here is the midgut. Okay. Yes, don't talk to yourself. Listen here. So this is the midgut. Okay, this is the midgut. But students, more than a midgut, what is important? What is present between the foregut and the midgut? So there is a presence of 6 to 8 thin tubules which are called the hepatic CK. Hepatic CK. Understood? Hepatic CK, and this hepatic CK can also be called as the gastric CK. Why? Because these people are going to help with the secretion of gastric juices. Okay, CK. So hepatic CK or gastric CK, which are how many in number? 6 to 8 tubules. Okay, 6 to 8 tubules. Done. That is present between the foregut and the midgut. As we come down, students, this is the midgut, or we can call it the mesentron. Okay, mesentron. Now, after you finish the midgut, you will automatically come to another region which is present between the midgut and the hindgut. So this is the hindgut. That is called as your Malpighian tubules, which you are going to study in your secretion again. What are they? Malpighian tubules. These Malpighian tubules are 100 to 150 in number, and they are yellowish tubules which are going to help with excretion. And then when we come to the down portion, which is the hind limb, you have ileum, colon and rectum. Okay, ICR, this together forms the hind gut. Any doubts so far? Any doubts so far? This is clear. The digestive system, you start from the esophagus. Okay, mouth has taken the food, it will go to the esophagus, esophagus to the crop to gizzard, where the grinding will happen between the foregut and the midgut. Okay, between the foregut and the midgut, what do we have? We have the hepatic CK or the gastric CK. Okay, hepatic CK or the gastric CK. Agni. 1 second, students. Clear? Okay, fine. So, esophagus, crop, sac-like structure, then you have gizzard, which is proventriculus, grinding will happen. Between the foregut and the midgut, you have hepatic CK or digestive CK, which is going to do the secrete the digestive juices. Now, what is the question going to come? How many in number? So, the number should be 6 to 8 tubules. 6 to 8 tubules, okay? Then, after the midgut, students, the midgut is also called as mesentron. You don't have to break anywhere, head for midgut. Then, it has to be the Malpighian tubules, which are 100 to 150 in number. Thin tubules which are present, yellowish in color, which are going to help with excretion. As you come down, you will see the hindgut, which is ileum, colon and rectum. Ileum, colon and rectum, and that is where the undigested food will go out. Clear to all of you? Any doubts in digestive system? Tell me now, Ali. Any doubts in digestive system? Quickly. So, what is the pathway of the food, students? What is the pathway of the food? Pathway of food, you start from the mouth, Okay, just to make an idea. Okay, you go to the esophagus, not you, the whatever the cockroach has it. Okay, esophagus, and then it will go to the crop where the storage of food will happen. Then it will come to the gizzard where the grinding will happen. So G for gizzard, G for grinding. Also another name for gizzard is proventriculus. They have asked this as a PYQ. From the gizzard, it is going to come to where? It will go to the midgut. Okay, it will go to the midgut. From the midgut, it will enter into the your hindgut. But before that, it will go to the ileum, then colon. Then the undigested food will come out through the rectum. Clear? The undigested food will come out through the rectum. In this, you have to only remember that to the pharynx, the food is going to, to the esophagus, the food will go through the pharynx. Done. Pathway is very clear to all of you. Yes or no? Okay, come on. Quickly. Come on, tell my God. Okay, this is going to do storage. Students, functions are important very much. Okay, storage. Then this is going to be grinding. Okay, and midgut, you know, mesenteron, ileum, colon, rectum also, you know. Only this much you have to remember. That's it. That's it is the digestive system. Okay, yes. Very good, very good. If you have already written, I did not see that. Okay. Now, circulatory system. Circulatory system of cockroach also is very simple, students. How primitive the organism, that simple their body functioning also. Now, in circulatory system, you have to understand that these people have heart, but the heart is muscular. Oh, the heart is muscular. But with the heart being muscular, it is also, we should understand, these people are elongated. Okay, elongated. Okay, and these people are present in the form of chambers. Okay, now how many chambers are present? Important. They have 13 chambered heart, and this 13 chambered heart is in position. Okay, is in position, and the relaxation and the contraction will be taken care by your alary muscles. What muscles, students? Alary muscles. Now, all of you tell me how many alary muscles will be there. If 12 chambers are there, how many alary muscles will be there? Quickly in the chats. So, let's consider this is the chambers. Okay, this is one chamber. Okay, like this one chamber is there. Then we have this alary muscle. Okay, let's say like a funnel shape it is. Okay, something. Oh, wait, I have to take it from the center. By then you answer this question, all of you. Something like this. Okay, yes. Yes, what's happening? Yes, 10 plus teen. Oh my God. From where do you people find such max? Okay. Yes. Okay, done. Clear to all of you? Okay, moving forward. Alary muscles, it's okay, students. They don't have to like. Okay, it's okay. Now you people come back to the class. Okay, don't worry. Yes, alary muscles are 12 in number. Okay, alary muscles are 12 in number because the last one, the last chamber will not have the alary muscles. They are 12 in number. 12 in number. Got it? 12 in number. Now, what is the function of alary muscle? Not mentioned in your NCERT, but if you want to really know it, it is for the relaxation and contraction. Okay, the muscles will help in relaxation and contraction. Clear? Relaxation and contraction. Done. Now, students, here one more thing that you have to understand. They have small spaces or like more than like a pore system, which is called the ostia. Okay, which is ostia. So the blood will enter through the ostia. It will move upwards and it will move again upwards to the head sinuses. Okay, they have open circulatory system, so they have sinuses or spaces. So they will go to the head sinuses and again they will return back. Okay, so this cycle will keep continuing. Understood, all of you? This cycle will keep continuing. Okay, clear or not? Perfect. Okay, clear? It's okay, blue paper. Okay, 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 fine. Now moving forward. Now this you understood. So these people have what? They have open circulatory system. Open circulatory system means what, students? They do not have proper capillaries which will take the blood. So because of that, what will happen? The blood will go into spaces which are called as sinuses or the spaces. Clear? Sinuses or spaces. Done, done, done. Done, done, done. Yes or no? Perfect. Okay. Now look here, students. This is the structure. Now here you will see anterior aorta. This is the main aorta. Okay. This is the main aorta. So alveolar muscles are there. Chambers of the heart is there. So it will enter through small sinuses. It will go all the way to the head sinuses and it will come back. So if you take a cross section and see, this is how you will see, but you cannot see a lot of it, but you can see the dorsal vessel like this. Okay, dorsal vessel, and it is running through the mid dorsal line. Okay, this entire heart is going through the mid dorsal line. Mid dorsal line. Yes, done. Okay. Yes, perfect. Perfect. Now, moving forward to... Okay, now students, we have to study, not reproductive system, we have to study this one, your nervous system, right? So here I am going to write that. Okay, here I am going to write that. Ready all of you? Any doubts? Any doubts? We have to study now excretion, okay, respiration, excretion, respiration. So I think we can take new slides only and write, no? Yeah, we'll write it here, then we will move it to the forward, okay? Okay, let's change the color. Color has got boring. One color, two color. Okay, come on. What is the system we have to study? I am from Alapi, okay, Alapura, boat house. All of you will know boat house, no? Very well, you will know boat house. You'll be like, ma'am, even I want to go to boat house. Students, don't worry, even I haven't gone to boat house. Okay, done, done, done. Okay, fine. Now here, when we talk about the circulatory system, one very important thing that you have to remember here is, okay, I'm deleting this, 1 second. Which slide have I selected? Okay, 1 second, students. Okay, see, when you learn about the circulatory system, you have to remember one thing, that out of the 13 chambers, okay, out of the 13 chambered heart, 3 of them are in the thorax. Okay, 1, 2, 3 is in the thorax and the rest 10 are in the abdomen. So this number 10 is very important for the cockroach. Okay, even though it is not important for you, for the cockroach it is very important. Okay, 3 in the thorax and 10 in the abdomen. Now when we go to few other systems, students, let's divide the page. Okay, let's divide the page, because there's very less about these systems. Okay, there's no much about these systems. Okay, we will start with the respiratory system. Respiratory system. Now, respiratory system, what do you know? See, students, this is the cockroach. Okay, it's a beautiful pink cockroach I have drawn. Now, if I like, let's say I'm drawing some spaces. I know you can see the black mark, but still, act like it is spaces, okay? I told you, whatever I draw, you accept it. Yes, these are the spaces. Can you see? Now, what is going to happen? What is going to happen? These spaces are called as spiracles. What are these spaces called as? Spiracles. Spiracles. And through the spiracle, students, the air will enter inside. Okay, the oxygen will enter inside. And this, to control these spiracles, okay, how many spiracles are there? Ma'am, there are 10 spiracles. Okay, there are 10 spiracles. Out of this 10 spiracles, again classification, 2 will be in the thorax, 8 will be in the abdomen. 8 will be in the abdomen. Clear to all of you? Yes or no? So the spiracles, the spiracles will be the small openings through which the air can enter inside to the cockroach body. Now students, the spiracles has to be controlled, right? If the spiracles are not controlled, all the time exchange will happen. We do not want that. To make sure that there is a control in the openings, okay, in the spiracles, we have something called as spinsters. We have something called as spinster muscles. All of you know spinster muscles. I've taught you this in excretion. So we have something called as spinsters, which will regulate the opening and closing of the spiracles. Okay, it will regulate opening and closing. Clear? Opening and closing of the spiracles. Any doubts so far? Any doubts so far? If you have doubts, you should ask now. No doubts? Okay, good job. Good job. Now, students, other than this, these people also have one more organ for excretion. Okay, one more organ for excretion. What is that one more organ? For respiration is the presence of tracheoles. Okay, it's the presence of tracheoles. So, this is your tracheal tube. This tracheal tube ends in a, like a small network of tubes. Okay, networks of tubes which are called the tracheoles. And these tracheoles are going to help with the respiration. Clear to all of you? That's all. Someone was telling, ma'am, this is so difficult. What is so difficult? Nothing is difficult. So these are your tracheal tubes. They end in small networks called as tracheoles. Okay, tracheoles. Clear? In arthropoda also we have studied that the respiration happens with the help of book lungs, book gills, okay, and with the help of tracheal systems, right? So this is the thing. Clear? Now, when we talk about excretion also nothing is there, students. Excretion, you already studied one term. What was that one term that you already studied? That, ma'am, these people, that is between the midgut and the hindgut, these people have a structure which is called what? Which is called the Malpighian tubules. Which is called the Malpighian tubules, which are 10 to 150 in number. Okay, 10 to 150 in number. These people are yellow in color. Okay, they are yellowish in color. And these people are going to be the main organ for excretion. Done, yellowish in color. Now, what you have to study additionally is that these people have some additional organs. Okay, additional organs which are going to help. Okay, students, tell me if they are uricotelic or urotelic. Okay, they are uricotelic or urotelic. All of you tell me in the chat box quickly. Fast. Are they going to give uric acid or urea? So the additional organs, by the time you tell that, I will write this. Additional organs are going to be your fat body. Okay. Uricose gland and nephrocytes. Very good, very good. Ma'am, they are nitrogenous waste. They are going to absorb the nitrogenous waste and they are going to give out as uric acid. Okay, uric acid, and because of that, these people are called what? These people are called uricotelic organisms. Okay, they are uricotelic. Done. Now, here students, one additional, like an important thing that you have to study is about your uricose gland, which is only present in the male. And this uricose gland is made up of your long tubules, and these long tubules are a part of your mushroom gland. Correct? So, remember, uricose gland is only present in males. Okay, done. Okay, this is your excretion. Any doubts in excretion? Next, your neural system or your nervous system. Some of you might find this very difficult, but it's not that difficult, students. It's just a small thing. Okay, it's just a small thing. Now, look here. Your nervous system, when I talk about, okay, nervous system, when I talk about, understand these people do not have well-developed system, but they do have network. Okay, network of cell body together to form ganglia. Okay, or we call it ganglion. Now, you tell me this, if this is the esophagus, okay, if this is the esophagus, something that is present top of the esophagus or above the esophagus, I will call it supra. Okay, I will call it supra. So here also, same thing is going to happen. Since it is present above the esophagus, there is going to be a supra esophagus or phageal, okay, ganglion, which is going to act as the brain, which is going to act as brain. Clear to all of you? Which is going to act as the brain. Now, ma'am, what is this brain going to do, ma'am? It is going to supply, okay, it is going to supply nerves to the eyes and antenna. So, a ganglion, okay, a group of cell body which is going to form or act as the brain, which is called the supraesophageal ganglia. This is difficult. Sorry, this is difficult to remember. No, right? Okay. Now, students, before I go this whole thing, I told you these people have ganglion, but how are they present? These people are present in series of, okay, series of segmented. They have a series of segmented ganglions which are connected. Okay, they have to be connected. No, they are connected or attached, I can say. If I use the word connected again, connected, connective, you will get confused. Attached with the help of longitudinal connectives. Connectives word only you can understand, it's going to connect something. So with the help of longitudinal connectives. Clear to all of you? Yes or no? Any doubts? Okay. So brain is represented by a supraesophageal ganglion, which is going to supply the nerves to the antenna and the eyes. Other than that, we understand that these people have a series of segmented ganglions which are connected by the longitudinal connectives. Okay, easy peasy lemon squeezy. Okay, now being said that, can we come to reproduction chapter? Can we come to reproduction, sorry, not chapter, topic. Can we come to reproduction topic? Reproduction is very easy. I want your 5 minutes attention to this. 5 minutes attention to this, you will understand this. Yes? Well, you people are roasting each other only. You don't need me, no? Okay? Yeah, supra, you will remember with your friend Suvra. Okay, but in the examination, don't see that, oh, Suvra is not there, so I will not mark the answer and come off in the exam. Okay? Yes. Yes. Now look here. Cockroach are dioecious. What is dioecious? Dioecious means, ma'am, sexes are separate. Sexes are separate. Right? Sexes are separate. Now, both sexes have well-developed reproductive organs. Now, male reproductive organ consists of a pair of testes, one lying on each. Now, question has come from here. Lateral side of the 4th to the 6th abdominal segments. Lateral side, okay, 4th to the 6th abdominal segments, okay. Now, from each testis arises a thin, vas deferens which opens into the ejaculatory duct. Through what, students? Through the seminal vesicle, who is going to do the job of storage. Right? Now, the ejaculatory ducts opened into the male gonophore. I told you male gonophore is the opening, okay, which is situated ventral to the anus. So, dorsal anus is there, ventral, the gonophore will be present. Now, a characteristic mushroom-shaped gland. Mushroom-shaped gland is present, okay, in the 6th to the 7th abdominal segment, which functions as an accessory reproductive gland. That means it will do some other functions, okay, some other functions like your prostate gland, Cowper's gland and all of that. Okay, now the external genitalia are represented by the male gonopophysis or the phallomeres. Okay, they are represented by the male gonopophysis or the phallomeres, which are asymmetrical chitinous structures which are also going to protect the male gonophore. Okay, now students, the external genitalia are represented by male gonopophysis or phallomeres, and the sperms are stored. Important, very important. S for sperm, S for storage, S for seminal vesicle. Sperms are stored in seminal vesicle, and I told you this, sperms are not going individually, they are going as a packet, they are going as a bundle. Clear to all of you? They are not going as a packet. Clear? Yes or no? They are going as a packet, they are not going as individuals, they are going in a form of packet and these bundles. And these bundles are called what? These bundles are called as the spermatophores. These bundles are called as the spermatophores, which are discharged during copulation. Okay, now let's learn the structure, then I will make you write. Now look here. We start with the first thing, that these people have a pair of testes, right? These people have a pair of testes. So here, pair of testes. Tell me the segment they are present from. They are present from which segment to which segment? Quickly in the chat box. Quickly in the chat box. And students, when you look at this pair of testes, no, I want all of you to focus here. This testis is trilobed. Can you see? Like this, 3 lobes are there. These people are trilobed. Trilobed. Where is it present from? Yes, very good. From the 4th to the 6th segment. Okay, 4th to the 6th segment. Now, what did they say? They told that your testis is going to open into a small tube-like structure which is called the vas deferens. So, if you write the flow chart from testis, you will write vas deferens. And then they told that from the vas deferens, it is opening into the ejaculatory duct through the seminal vesicle. Okay, through the seminal vesicle. Seminal vesicle is going to do what? Storage, ma'am. So, it is going to open into the ejaculatory duct through the seminal vesicle. Done, done, done. Now, we are learning everything that is present in the diagram, okay? Then they told us that there is a presence of an accessory organ which looks something similar like a mushroom. So, we call this as the mushroom gland, which is made up of small tubules, long tubules and seminal vesicles. So, remember like this, your entire mushroom gland has 3 components, that is your small tubules, long tubules and your seminal vesicles. So, short tubules, long tubules and the seminal vesicle. Clear to all of you? Clear to all of you? Yes. Now, look here. Now, look here, students. This, on top of the mushroom gland, you have one more gland which is called the phallic gland. Now, ma'am, what will this phallic gland do, ma'am? I will tell you in the next slide. Okay. Now, when we try to understand the diagram, we will see right phallomere, ventral phallomere and left phallomere. These are asymmetrical chitinous structures which are going to protect the, or which are going to represent as the external male genitalia, at the same time protect the male gonophore. So, students, these 3 words are clear to all of you. Now, anal cirri, you know. Caudal style, also known as anal style, also you know. Now, pseudopenis, okay, as the name itself suggests, it is pseudo, false, okay, false. Now, students, you have titillators. This is actually going to help in copulation. These people are going to help in, release of the spermatophore, and also for during the copulation to stay in a position and all of that. Okay, the titillators are going to do that. Is there any word in this diagram that you don't understand? Is there any word in this diagram that you don't understand? Please tell me in the chats. Please tell me in the chats. No, right? No, right? Pakka? All of you? I can only see few of them answering. So, students, here I told you that there is a pair of testes, 4 to 6 segments. You understood? So, let's also write the pathway. I'm telling. Okay, the passing of the sperm. Okay, so it starts from the testis. It is formed in the testis. From the testis, it will come through a tube which is called the vas deferens. From the vas deferens, it will come to the ejaculatory duct. Okay, ejaculatory duct through the seminal vesicle. From ejaculatory duct, where will it go, students? It will go to the male gonophore. Okay, male gonophore. Now, other than this pathway, you need to understand that this sperm is not going. This sperm is not going single like you people. Okay, it is going as a packet. Okay, it's going as a packet. Now let's take an example. This is the packet of sperm and I call it the spermatophore. Okay, something is wrong with the spelling, no? Sorry, yeah. Spermatophore. Now this spermatophore needs a proper covering because it is going to another war, right? It is going outside, it's going for a battle. So we need to give the protection. So here what is going to happen, students? We are going to give an outermost coating, innermost coating, and this outermost coating is done by your phallic gland. This outermost layer is done with the help of phallic gland. Clear to all of you? Yes or no? Yes or no? Okay? Done. Is this clear? Is this clear? That your spermatophore, the outermost layer, is made up of your phallic gland. Okay, and the spermatophores are glued, right? Glued together. They are not going as I told you. So they are glued together and they will be going to the female. Done. Okay. Yes. You want me to write that? Now here it is 4th to 6th segment. You'll make me write everything. You people will write nothing. Okay. Then with this you also have an additionary gland which is the mushroom gland. Okay. And this is going to be present from the 6th to the 7th segment. 6th to the 7th segment. Now going to the female one. Okay. Yes. So look here, students. The female reproductive system consists of 2 large ovaries lying laterally, again laterally, from the 2nd to the 6th abdominal segments. Now tell me the answer. Ovary is bigger or this testis is bigger? Ovary is bigger or testis is bigger? Quickly in the chats. Each ovary is formed of a group, very important. Each ovary is formed of a group of 8 ovarian tubules. In other words, you can also call it the ovarioles, containing a chain of developing ova. So each of the ovarioles will have the mature ova and then there will be developing ova. So if you consider each ovaries can make up to 8 mature eggs. 8 mature eggs. Okay. Yes, ovary is bigger. Very good, because it is present from the 2nd to the 6th segment. So it is bigger. Now students, containing a chain of developing ova, oviducts of each ovary unite into a single median oviduct called as the vagina. Clear to all of you? Which opens into the genital chamber. Done. A pair of spermatheca is presented in the 6th segment. Students, you might be thinking, ma'am, are you reading it correct? Yes, a pair of spermatheca. Why has this word come in the female reproduction? Because there a packet of sperm is coming. No, to receive that someone should be there. So for that receiving, you have spermatheca in the female body, which is present in the 6th segment. So there you saw mushroom gland from 6th to the 7th segment. Here you have 6th segment, your spermatheca. Okay, which opens into the genital chamber. And genital chamber has 2 parts, genital pouch has 2 parts, vestibulum and genital chamber. Okay, yes. Now sperms are transferred through, see, understood? Sperms are transferred through the spermatophores and their fertilized eggs are encapsulated. Okay, that means you have seen the cockroach egg, no? They have a coating and that egg we call it as the ootheca. The coating is done by a collateral gland. Okay, there it was phallic gland, here it is collateral gland. And ootheca is dark, reddish, blackish, brown capsules and they are dropped into small spaces or the cracks where they get ample amount of food and moisture. Okay, so a female can produce 9 to 10 ootheca, each containing 14 to 16 eggs. That means up to 160 fertilized egg these people can release. Now, the development of Periplaneta americana. Okay, we will come into that. So, students, any doubt in this? First, let's learn the structure. This is your female. Okay, reproductive system. Now these are the two ovaries. Now in the ovaries, if you see, there are long tubule-like structures which are called your ovarian tubules or ovarioles. What are they called? Ovarian tubules or ovarioles. These ovarioles will have your developing ova. Developing ova. So students, if you assume that each of them gives a matured egg, then this ovary can give 8, this ovary can give 8. Okay, that is why they give 14 to 16. Clear? Now, ovary you understood? Yes, ma'am, you understood the ovary. Which segment to which segment? 2 to the 6th segment. Then oviduct is there. Yes, ma'am, we understood the oviduct. Now, here is where your confusion will come. They will ask you to label oviduct and common oviduct. So here, don't get confused. Look at both the oviducts are opening into a point. That is called the common oviduct, or in other words, called the vagina. Now, from there down, if you see, students, you will see this tree-like structure which is your collateral gland. Now, this collateral gland is going to make the outermost layer of the ootheca. Okay, ootheca. The outermost layer of the ootheca will be made. Now, as you come down, you notice that there is a genital chamber and there is a vestibulum together called the genital pouch. This is where the egg will be there. Can you see how the structures are? Like this. Okay, the egg will be inside that, and then obviously these people also have gonapophysis, and here the most important spermatheca, which will be receiving the spermatophore, which will be receiving the spermatophore. Spermatophore. Done, done, done. Okay. Oh no. Okay, clear? Yes or no? Any doubts? Anything doubts? Okay, now here students, another important thing, the cockroach develops in porometabolous. Okay, development is porometabolous. That means what? These people are going to grow in nymph stages. Okay, they will go through nymph stages. How many nymph stages they will go through exactly? They will go through 13 times. Okay, 13 times they will go through the molting about 13 times, and only in the last second one they will start seeing the wing pad. Last second one they will start seeing the wing pad. Okay, so cockroaches have wings, that is only the adult cockroaches. The last nymphal stage will have the wing pad. Clear? Will have the wing pad. Okay, students, their benefit is not known. They are mostly useless to us. Okay, they are mostly useless to us. Is this clear to all of you? Yes or no? Any doubts so far? Any doubts so far? No, right? No, right? Can we start frog? Can we start frog? Can we start frog? Yes, 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 yes. Crocodile, sorry. Because of this people only, they have written cockroach spelling like crocodile. I'm looking at that and telling. Cockroach is done, done. All of you put it in, all of you type that in the chat box. Cockroach, done, done. Quickly in the chats. Cockroach, done, done. Jaldi, jaldi. I'll go fill my water. I'll get some water, students. By then, you people prepare yourself. If you want to write down something, make a note of it, okay? Then we will start cockroach. Come on. Yes, ready? Ready, steady, go. Wow, so much excitement for frog, yeah? Frog should know this, that frog has a fans association. Okay? I'm teaching NEET students from past 2 years. No fans only for me. Look at this frog. Okay? Look at this frog. Full fans. Papa, NTL is still confused with frog. Is it an insect or an amphibian? Okay. Come on. Yes, frog is looking that side. It's not interested. Okay, so we have to make it interested. Yes, look here, students. Animalia, obviously phylum is going to be Animalia. Superclass Tetrapoda. Now we have learned this before also, that when we did the categorization of your vertebrata, we studied Egnatha and Gnathostomata. In that we studied that depending on the presence of depending on the presence of fins, okay, fins, and depending on the presence of limbs, we did the category. That is, fins we divided to Pisces and limbs we divided to a superclass called Tetrapoda. Okay, from the term itself you understand tetra means 4, poda means leg. Okay, poda means leg. So here Tetrapoda class Amphibia. Amphibia means what? They can be in two life forms, that is on land, okay, any two kind of bios, okay, land and water. Land and water. Now Anura, students, it's coming under order Anura because of the presence of their jointed legs. Okay, because of the presence of their jointed legs. Clear? Clear? Okay, I have hands. Yes. Oh, don't get scared of frog. It's hardly coming out in the breeding season. That time you will be inside the house. Right, frog will be sleeping during the peak summer and peak winter. It comes out only during the breeding season, that is the rainy season. That time you will be inside the house. Why will you get scared of something that you will never meet in your life? Tell me. Come on. Don't be scared of something that you will never meet in your life, right? So, genus is going to be Rana tigerina. This is the one that we are going to study. Okay. Yes, Indian bullfrog, we are going to study about this Rana. This is our Rana tigerina, which is turning that side and putting full head weight and looking that side and sitting. Okay, because it knows that we are going to study about it and 4 marks will come from this. So, ahankara. Okay. Clear? Yes. Okay, let's start. Let's start. What do you know about frog? Okay, what do you know about frog other than making croak croak sound? Other than making croak croak sound. Okay. Yes. Now, frog, students, general characteristics, you know how the frog looks, no? That is, these people have, their skin is moist and slimy. Okay, their skin is moist and slimy. Second thing that we know is the dorsal side. Okay, there is the dorsal and the ventral side, no? So, the dorsal side has olive green. It is olive green in color, okay, with dark spots, okay, with dark spots. Then ventrally, okay, ventrally they are yellowish in color. Okay, they have an yellowish color. Clear to all of you? Yes or no? These 3 things generally you have seen, no? Yes? Okay. Okay, perfect, perfect. Okay, very nice. Now, students, olive green with dark spots, ventral yellowish in color. These people are poikilotherms. What are they? Poikilotherms. What is poikilotherms? Ma'am, poikilotherms, other word is cold-blooded animals or organisms. That means what? They will not be able to maintain a constant temperature, but they will keep changing the temperature with the external environment. Okay, their temperature will be changing depending on the external environment. Clear to all of you? Important. Okay, important. Yes. Anything else you know about frog? Anything else you know about frog? Come on, quickly tell me in the chats. What else do you know about frog? You know that they can live both on land. Okay, they can live both on land and water. This also you know. And these people have different respiratory systems, right? They have the skin, they can respire through the buccal cavity, they can respire at the same time with the lungs. Okay, these are the things that you have to know about frog. Is it so difficult? It's not very difficult. Okay, now students, these frogs have 4 limbs. Now 4 limbs. From that only you remember these people are 4 in number. Okay, this has come in one of the PYQs, 4 in number. Then they have hind wings. They have whom? Hind wings, which is 5 in number. And most importantly, they are webbed. What is the meaning of webbed? What is the meaning of webbed? Now let's draw this. Okay, this is the leg. Let's assume. Okay, now this is webbed. What are they going to use it for? They are going to use it for swimming, jumping, all of this they will use it for. Okay, these people have webbed structures. Okay, webbed foot, and these people are going to help in swimming, jumping, leaping, everything. Okay, now body of frog. Okay, body of frog is divided. Body of frog is divided into two things, students: head and trunk. Head and trunk. Ma'am, that's so sad, ma'am. Only two parts are there. Why? Because they do not have the neck or the tail. Neck or tail is absent. Why, students? I have told you this. If the frog has to leap, okay, the neck will become a problem. So the frog will be like, hey, why to chumma waste energy? Let's not have a neck. Okay, so neck. So when they jump, their neck will be a problem. So these people don't have a neck. Okay, they leap. They take big heights. No? Yes. And students, this frog, they have 3-chambered heart. 3-chambered heart. That means mixing up of blood will happen. Okay, 3-chambered heart. Mixing up of blood will 100% happen here. Okay. Now, other than the body, what do you know? These people do not drink water. Why? Because they absorb everything with the help of their skin. And their skin is always kept slimy. Okay, slimy and moist. Why? For diffusion to happen. Okay, for diffusion to happen. Now, respiration here is going to be of 3 types. Okay, they can respire with 3 organs. What are they? As I told you, skin. Okay, they will use the skin. Okay, lungs and buccal cavity. Okay, buccal cavity. Mainly we say skin and lungs. Okay, buccal cavity also is there. So skin, students, if the respiration is happening with the help of skin, we call it cutaneous respiration. We call it cutaneous respiration. Okay. Now, if it is lungs, what will you call it? Pulmonary. Whenever lungs come, always use the word pulmonary. Pulmonary respiration. Okay. When it is buccal cavity, it is called as buccopharyngeal. Buccopharyngeal cavity. Sorry, respiration. Buccopharyngeal respiration. Any doubts? Any doubts? Okay, understood? Yes. Oh yeah, hind wings I wrote, no? Sorry. Okay, yes. So we will learn more about it. I have put NCERT highlights, okay, just to make everything more thorough and clear for you. So body of frog is divisible into head and trunk, okay, head and trunk, and a neck and tail are absent. Okay, as I told you, a neck and tail are absent. Above the mouth, now very important, students, above the mouth, there is a pair of nostrils. Okay, I'm not the frog, so you cannot imagine me as a frog. Okay, mouth, above the mouth, nostrils, and above that eyes is there, and side to the eyes you have tympanum. Okay, understood? Like that only. So above the mouth, a pair of nostrils is there. Okay, a pair of nostrils is present. Eyes are bulged and covered by a nictating membrane. What is the job of nictating membrane? To protect them underwater. Okay, to protect them under water. On neither side of the eyes, important, on neither side of the eyes, they have the tympanum. Okay, last time also I tried drawing a frog, but it was a very big fail. Students, put that other, you people have this emoji. Yeah, this emoji is not good. Okay, so let me look at a frog so I can draw a frog. Okay. Am I going to be on every platform for drawing this frog like this? No, I can't draw frog, students. I've given up on frog. Okay, I can't. I used to draw frog, okay, for my masters and all, but now I'm not able to draw. So just assume that this is the eyes. There will be a membranous structure called the tympanum, and front of this there will be two nostrils. Clear to all of you? That's why I wanted to draw this. So like this one, kutti nostrils will be there. Eyes. This is the eyes. Okay, I'm putting eyelashes just to understand, and then a membranous structure called the tympanum. Clear to all of you? Yes or no? Okay, let's not see the frog again. Yes. So yes. Okay, there. First draw the body. Face only I'm not able to draw. What I'll draw with the body? Body, if I draw, it will come or what? It will not. Let me try. Since my students are telling. Does it look like a frog? Huh? With which angle it looks like a frog? Okay. Ninja. Don't ask like that. Okay, don't ask, ma'am, what is this? Okay, I told you people. If I say it's frog, it's frog. That's all. Over. Conversation over. Okay, your mom tells you, no, this is chapati, even if it doesn't taste like chapati, just like that only. Okay, now, keeping this in mind, you understood. Okay, tympanum is there, eyes is there, and nostrils will be there. Okay, the forelimbs and the hind limbs help in swimming, walking, leaping, and burrowing. The hind limbs ends in 5 digits. Important. So I told you 4, you remember, 4 limbs, 4. So the other one will be 5. Okay, and they are larger and muscular than the 4 limbs that end in 4 digits. Feet have webbed, I told you, no, webbed digits. Why, students? To help them in swimming. Okay, webbed digit to help them in swimming. Clear? Yes, thank you. It looks like a frog. Yes, ma'am. Who told dinosaur? I will send you with dinosaur only, okay? I will send you with Basava. Fine. Now, lymph. Yes, lymph. Okay. Now, frogs exhibit sexual dimorphism. Very, very important, students. Frogs exhibit sexual dimorphism. That means male and female will have some special characteristics to differentiate the male and the female. What is the most important characteristics? The frog, okay, the frog will have a, that is the male frog will have a presence of sound producing vocal sac. And also, they will have a copulatory pad or else a nuptial pad, which is going to help them during copulation. Location is important. It is present in the first digit. Okay, this is the location. First digit of the 4 limbs. Okay, which are absent in the female frog. Clear? Yes or no? Yes or no? Any doubts? Any doubts? Okay. Now, let's understand the anatomy of frog, and obviously, we are going to start with the digestive system. For frog, I have highlighted NCERT because this is easier. If I write, no, the whole NCERT I have to write. Everything is important. So, here, look here. We will write the flow chart, okay? The digestive system of frog contains the alimentary canal, okay, and the digestive glands. Now, students, the alimentary canal is short. Why is the alimentary canal and the intestine reduced in frog? Because these people are carnivorous animal, and for carnivorous, the alimentary canal or the intestine does not have to be long as in herbivorous animals. Okay, so here the alimentary canal is short because the frogs are carnivorous. It is a statement based question. Okay, statement based question. Now moving forward. And hence the length of the intestine also is reduced. Hence the length of the intestine is also reduced. The mouth opens, important, same like how we think of our body, you know, same thing you have to remember here. The mouth will open into the buccal cavity. That leads to the esophagus, again through pharynx. Esophagus also is a short tube. Esophagus also is a short tube. Understood? Okay. Yes, and it opens into the stomach. It opens into the stomach, which in turn continues as the intestine and the rectum, and then finally it opens outside with the help of cloaca. Now, students, here you have to remember some more important things. Liver will secrete the bile, and the bile will be stored in the gallbladder. Pancreas will make the pancreatic juice and keep it with themselves. Now, what they will do? The undigested, the partially digested food will come as chyme to the stomach and it will go to the intestine. That time the common bile duct will give the bile and the pancreas will give the pancreatic juice, and then the digestion will continue. Clear? Understood? So here what is going to happen? Liver secretes bile that is stored in the gallbladder. Pancreas, a digestive gland, produces pancreatic juice. Any doubts? Any doubts? Okay. Now, students, the food is captured by the help of a bilobed tongue. We also say prehensile, no? But prehensile is not absolutely right because the frog tongue is not completely prehensile, but we can use the word prehensile. So, students, the tongue is bilobed, okay? The tongue is like this. Sorry. Like this, bilobed tongue. So, it will be helping in catching the insects. Now, the bilobed tongue, the digestion takes place. Remember here, digestion of the food takes place by the action of HCl and gastric juices. Just like in us, same thing happens. Secreted by whom? The walls of the stomach. Whatever I have underlined, only you have to read. Now, partially digested food called the chyme is passed from the stomach to the first part of the small intestine, that is the duodenum. And in the duodenum, what will happen is the duodenum will receive the bile from the gallbladder and pancreatic juice from the pancreas through the common bile duct. Understood? Through the common bile duct. Bile will emulsify the fat. Bile will emulsify the fat. That means the fats are hydrophobic to water. So, bile will go and attach and make an outer layer which is hydrophilic, so we can break down this fat. So, bile will emulsify the fat, and the other side, pancreatic juices will digest the carbohydrates and proteins. Clear to all of you? Yes or no? Yes or no? Okay. Okay. Now, final digestion takes place in the intestine. Digested food is absorbed by numerous finger-like projections called the villi. Okay. Inner wall of the intestine called the villi and the microvilli. The undigested solid waste will be passed into the rectum and then it passes into the cloaca. Please write this. Please write this. Yes. Understood? Understood? Okay. Now moving forward. Okay. Is this clear to all of you? Yes or no? Any doubts? Any doubts? Okay. Give me the flow chart. Give me the flow chart. So the food is taken in with the help of bilobed tongue. Now with the help of this tongue, the food has entered into the mouth. From the mouth, it is going into where, students? The buccal cavity. I want all of you to answer. Even if I am writing, it's okay. You keep answering. To the buccal cavity to esophagus. Okay, through pharynx. Now from esophagus, it is coming to the stomach. Okay, esophagus, it's coming to the stomach. In the stomach, the HCl and the digestive juices are getting secreted by, like through the walls of the stomach. Okay, from the stomach, the partially digested food that is called the chyme. Chyme is going where? To the duodenum. Okay, then in the duodenum, what is happening, students? This food has to be digested. Whatever chyme has come has to be digested. For that, one side we have liver. Okay, one side we have liver who has made the bile and given it to the common bile duct. Sorry, who has stored it in the gallbladder. Okay, stored it in the gallbladder. Okay, then we have pancreas. Pancreas that is giving pancreatic. Oh, I'm not able to write. pancreatic juice. Okay. Now, both of the secretions will be given to the duodenum with the help of a common bile duct. With the help of a common bile duct. Done. Common bile duct. Done, done, done. Now, the undigested food will go, okay, again, it will go out through the rectum. Okay, whatever absorption has to happen, it will happen with villi and microvilli, and from the rectum, it will go to the cloaca to cloacal aperture. Clear to all of you? Any doubts? Any doubts? Yes, listen to the class. Please focus. Stop talking to each other. Okay, yes. Okay, clear? This is your digestive system. Now, when we come to the respiratory system, students, respiratory system, as I told you, that much only is there, that where the frog is going to use the skin, lungs and the buccal cavity. Okay. Now, look here. Frog respires on land. Okay. Respires on land and in water by two different methods. That is, in water, the skin acts as the aquatic organ, respiratory organ, and that kind of respiration is called cutaneous respiration. Now, dissolved oxygen in water is exchanged through the skin by diffusion. So, underwater, the frog is going to use skin as a medium for respiration. Why? Because skin is slimy, moist, and also it was going to help with diffusion. Okay, now, on the land, the buccal cavity, the skin and the lungs, all the three are going to be used as the respiratory organ. That is, the respiration by lungs is called pulmonary. Just before I taught you, buccal cavity is called buccopharyngeal. So, he is going to use, that is, the frog is going to use all the three methods when it is on land. When it is on land. Okay, now, students, the lungs are pink. Okay, pair of elongated pink colored structure. Okay, pink colored sac-like structure which is present in the upper part of the trunk region. That is, air enters through the nostril. So, where will air enter? It will enter through the nostril, it will come to the buccal cavity to the lungs it will go. Okay. Now, during aestivation and hibernation, what is aestivation? Summer sleep. Hibernation, winter sleep. The gaseous, they are so lazy that they do not even want to use lungs or buccal cavity for respiration. They will tell the skin that you absorb the water, you do the respiration, I am going to sleep. So, during peak summers, they are going to sleep, that is called aestivation, and during peak winters, they are going to sleep, which is called as hibernation. Clear to all of you? Yes or no? Clear to all of you? Yes. What's up, bro? What's up? Everyone keep asking in the chat box. Okay, that's why they made what's up. For asking what's up, they'll come till this one. They'll come till YouTube. Clear? Okay. Okay. Good job. Now, yeah, very lazy frog. This is your respiration. Now, students, coming to a little complex topic, that is your vascular or let's say your circulatory system in frog. Okay, we are going to understand part by part. Don't get confused. Nothing is there to get confused. It is simple. I will write and make you understand later if you don't understand the paragraph. Okay. Now, the best way to study frog is NCERT. Because in NCERT, everything is there. There's nothing additional. But in cockroach, I feel the flow can be a little more better when we write and teach. Okay. But frog, it's always better to use NCERT. See, the vascular system of frog is well developed, closed type. There we had open type. Okay. Open type. Now, frogs have a lymphatic system. We also have a lymphatic system. These people also have a lymphatic system. They also have an entire vascular system. We also have an entire vascular system. Clear? Yes or no? So, look here. They have a lymphatic system. Okay. The blood vascular system also. Blood vascular system. Now, what does the blood vascular system have? What do we have? Heart is there. Blood is there. Blood vessel is there. Simple. Same thing here also. Heart, blood and blood vessels. Okay. Heart, blood and blood vessels. The lymphatic system has lymph channels and lymph nodes. Same thing we have? Yes, ma'am. Same thing we also have. Now, heart is muscular structure situated in an upper part of the body cavity. It has 3 chambers. Right atrium, left atrium, ventricles. So, mixing up of blood will happen in the ventricles. So, it has 3 chambers. 2 atria and 1 ventricle. 2 atria and 1 ventricle, which is covered by a membrane called as the pericardium. This also all of you know. Any doubt so far? Any doubt so far? No, right? So, a triangular structure. Now, from here starts a little bit, you know, some students can get confused, but there's nothing to be confused. Students, these people have some accessory structures which are going to help with the circulation. That is, first they will have a triangular sinus venosus, which will join to the right atrium. Now, when you hear the word venosus, you will be thinking ventricles, okay, something like that, but you write the opposite. That is, sinus venosus joins to the right atrium. Joins to the right atrium. Is this clear to all of you? Is this clear to all of you? Okay, fine. Now, it receives blood through the major veins called the vena cavas. Even our right atrium receives blood through the major veins called vena cava. Superior and inferior, here you will tell anterior and posterior. Okay. Now, the ventricles open into a sac-like structure which is called the conus arterios. So, arterios, atrium you will want to remember like that. So, write the opposite. Ventricles will open into this. Ventricles will open into this. Clear? Yes. Now, the blood from the heart is carried to all the parts of the body by the arteries. So, we will call it the arterial system. And all the blood from the body is collected and brought it to the heart. We will call the venous system. So, look here. The blood from the heart is carried to all the parts of the body by the arteries, arterial system. The veins collect blood from different parts of the body. Veins will collect blood from different part of the body and bring it to the heart and form the venous system. Okay. Now, special venous connection. Students, there is special venous connection between the liver and the intestine, and we call it, okay, we call it the hepatic portal system. What do we call it? Hepatic. I told you whenever liver comes, it's usually the word hepatic. Special venous system. What is venous system? Collecting blood from all the different parts of the body to bring it to the heart. In that system, there is two kinds of system, liver and the intestine, kidney and the lower part of the body. Anything related to liver, hepatic. So, hepatic portal system. Anything related to the kidney, renal portal system. Clear? Renal portal system. So, hepatic portal system and renal portal system. The blood is composed of plasma and cells. Okay, we will come into that. But anyone has doubt? Now, look at this diagram. This diagram will give you a clarity on sinus venosus and conus arterios. Okay. Now, look here. Yeah. So, if you look here, students, can you see this triangular structure? Yes, ma'am, I can see a full triangular structure like this. This triangular structure is called sinus venosus. This sinus venosus attaches where? To the right atrium. To the right atrium. Understood? Yes or no? Okay. This one. To this. Okay. To the right atrium. Now, here if you see, students, can you see one sac-like structure? This sac-like structure opens into, the ventricles open into a sac-like structure called the conus arterios or truncus arterios. Truncus arterios. Okay? Yes? Is the structures clear? Is the structures clear? Any doubt in this? Any doubt in this? Okay, here what they're saying is the blood is composed of plasma and cells just like us. We have RBCs, but the only difference here is the RBCs are nucleated. RBCs are nucleated and they contain red color pigment called hemoglobin. WBCs are there, platelets are there. Heart is the pumping organ. This much all of you know, right? This much all of you know. The blood will carry all the nutrients, gases and water, and it will take it to the respective sites. Here again, lymph is also given, students. Look here. The lymph is different from blood because it lacks few proteins and RBCs. Same as what we have studied in our body. Same as what we have studied in our body. All of you are telling no doubt, so I am assuming there is no doubt. Okay, there is no doubt. Now, excretory system. Excretory system, 1 minute break, okay? For me to just, my leg is hurting, just 1 minute, okay? Okay, just revise. Let me ask you all a question. What is the triangular structure that connects to the right atrium? Quickly in the chat box. Quickly in the chat box. Come on, quickly. Yes, very good. Sinus venosus. What about the sac-like structure? Ventricles open into sac-like structure called as conus arterios or truncus arterios. Now, excretion. The elimination of nitrogenous waste is carried out by a well-developed excretory system. The excretory system consists of a pair of kidney, ureter, cloaca and urinary bladder. Now, ma'am, what is so different from us? Cloaca. We say kidney, ureter, urinary bladder, right? These people have cloaca, cloacal aperture, opening, okay? Now, these are compact, dark, red, bean-like structures situated in the little posterior in the body cavity on both the sides of the vertebral column. Each kidney is composed of several structural and functional structures called as uniferous tubules or nephrons. My God, since Basava Sir scared me. I got scared because I assumed that you left.
No.
Students. Exactly. I thought Sir left, so I was not expecting anyone to enter the studio. Suddenly, someone just comes in. I'm like, okay, yes. So, students, there is structural and functional units called uniferous tubules or nephrons. Two ureters emerge from the kidney in the male frog, and the ureters act as the urinogenital duct. That means what? The excretory substance, urine as well as the sperm is coming out through one passage that is called the ureter. So, we call that ureter as urinogenital duct. Okay, which opens into the cloaca. But females are not like that. Females are like, I am independent. I don't want all this ureter's help. So, I will have an oviduct that separately opens into the cloaca, and ureter will separately open into the cloaca. Similar as in, humans. Clear to all of you? So, who is independent here? Females are independent. They will have an oviduct that will separately open. Separately, it will open into the cloaca, and a thin-walled urinary bladder is present in the ventral to the rectum, which opens into the cloaca. The frog excretes urea. Cockroach was uric acid. So, this is uricotelic animals. Done, done, done. Okay, done, done, done. Any doubts? Any doubts? Tell me quickly in the chats. Okay, so clear? So here what is happening? They have kidney, okay, they have kidney, ureter, urinary bladder, cloaca, cloacal aperture is there. Now students, what is going to happen? Kidney is a dark reddish color structure. Now, the kidney will have ureter open into the urinary bladder, and then there will be a cloaca or cloacal aperture. Okay, you have all seen the structure, no? Now, they are saying that in males, the ureter is going to act as a urinogenital duct. That means from the testis, whatever sperm is formed, it's going to come to the vasa efferentia. From there, with the help of Bidder's canal, it is going to come into the urinogenital duct and it is going to come outside. But here in females, they are like, no, we have an oviduct. Okay, oviduct. Clear? Yes or no? Yes or no? Fine. Now let's learn the nervous system. Nervous system, students, they have the CNS. Okay, they have the CNS, PNS, ANS. They have the CNS, PNS and ANS. That means what? Central nervous system, peripheral nervous system and autonomic nervous system. Under central nervous system, you have the spinal cord and the brain. In peripheral nervous system, you have the cranial nerves and the spinal nerves. In frog, it is 10 pairs. PYQ, 10 pairs of spinal nerves and cranial nerves. Okay? Yes or no? Fine. Now, ANS, sympathetic and parasympathetic. Now, look here, students. Okay, from where did it get skipped? So, here, students, brain is inside a bony structure called the brain box. Okay, brain box. It's still lagging. Still lagging. 1 second. Why always in my head diencephalons are there? Oh God, how many lags? Okay, it'll be fixed now. Don't worry, don't worry. How many lags? Okay, it'll be fixed now. Don't worry, don't worry. You know, it's an internet issue at this time. Yesterday also at this time internet issue was there, no? 1 second. Yeah, it's fixed. Okay, it got skipped again. Okay, it'll be fixed now. Don't worry, don't worry. Okay, it'll be fixed now. Don't worry, don't worry. You know, it's an internet issue at this time. Yesterday also at this time internet issue was there, no? Surya putting nazar emoji. Okay, paired cerebral hemispheres, unpaired diencephalon. Now, the midbrain is characterized by a pair of optic nerves. A pair of optic lobes, okay? Now, hindbrain consists of cerebrum and medulla oblongata. Now, students, see, it will be difficult for you to remember all of this. So, how you can remember? Still lagging? Now, okay? Yeah. So, brain is divided into forebrain, midbrain, hindbrain. Okay? Now, forebrain, you will remember like this. Forebrain, OCD. OCD, all of you know, no? OCD, all of you know, right? O for olfactory lobes. C for cerebral hemispheres. Okay? D for unpaired diencephalon. Unpaired and paired, you have to remember. Done? Diencephalon. Okay, OCD. Now, midbrain, you just have to remember. Midbrain, you have to remember mole. M-O-L. That is midbrain optic lobes. Okay. Then hindbrain. Okay, I'll write here. Okay. Now, hindbrain. How will you remember? Suvra, tell me. Hindbrain. Yes. Hindbrain. HCM. HCM. Okay. HCM. Like LCM, no? HCM. So, hindbrain has what? H for what? C for what? Hindbrain. Okay. Cerebellum, medulla oblongata. Okay. Medulla oblongata. So, this is how you will remember the parts. Here, the structure is there. Look at the structure. Olfactory nerves are there. Olfactory lobes are there. Olfactory lobes are present where? In the forebrain. Okay. Cerebral hemisphere is there. Fissure is there. And the pineal body, okay. Diencephalon is there. This together makes your forebrain. Then you have optic lobes. That is the midbrain. Then when you come down, medulla and cerebellum, that makes the hindbrain. Clear to all of you? Clear to all of you? Okay. If you're understanding and you're not able to solve the question, that means your practice is very bad. Okay, you need to practice more. You need to practice more. Students, control and coordination, nothing is there. Just all the endocrine glands you have to write. Okay, these people have the endocrine glands. You have to write that. Okay, endocrine glands. Okay, all the endocrine glands that is given in your NCERT. But again, question comes very rare from this topic. Okay, very rare from this topic the question will come. Now, moving forward to your reproduction. Look here, reproduction in frog, students. First, I will show you, first we will read through this, then we will go to the, diagram. Look here. Frog male reproductive organs consist of a pair of yellowish ovoid testes which are found adhered. Adhered means what? Attached. Okay, to the upper part of the kidney by double fold of peritoneum called as the mesorium. So, kidney and testes have a connection and they are adhered. Okay, oops. Did the mic have an issue? They have adhered with the help of peritoneum, which is called the mesorium. Vasa efferentia, students, they have the presence of vasa efferentia that are 10 to 12 in numbers that arises from the testis and they open to the kidney side and open into Bidder's canal. Now all of you will ask me, ma'am, where is Bidder's canal present? Bidder's canal is present in the kidney, but they are going to help with reproduction for transferring the sperm from the testis to the urinogenital duct. Okay, yes. Okay, thank you. Opens into the Bidder's canal. Finally, it communicates with the urinogenital duct that comes out of the kidney and opens into the cloaca. Understood? So here what is happening, the cloaca is a common opening for your digestion, reproduction as well as your excretion. That means the fecal matter, urine and the sperm is coming out of the same thing. Okay, the cloaca is a small median chamber used to pass the fecal matter, the urine as well as the sperm to the exterior. Now let's look at the structure and understand. Look at the structure, students. You will understand everything very easily. Now look here. This is your kidney. Okay, which is your kidney? This is your kidney. Now the kidney, to the kidney, who is attached? A ovoid yellowish structure called the kidney is attached with the help of mesorium. Understood? That is a peritoneum, double fold peritoneum called as mesorium. Now, in the kidney, there is something called as Bidder's canal. Okay, Bidder's canal. That Bidder's canal is going to help in the transfer of sperm. Now, look here. Fat bodies. Okay, that you leave it. Now, kidney, Bidder's canal. Now, from the kidney, it is going to come. See this, can you see? This is your vasa efferentia. Here, see, vasa efferentia. So, testis made the sperm. Sperm came through the vasa efferentia, then it went into the Bidder's canal. From the Bidder's canal, it will come into the urinogenital duct. This urinogenital duct will open where? Can you see the opening is it to the cloaca? From the cloaca, it will come out through the cloacal aperture. Done. Okay. Any doubts, students? Any doubts? Now we will study female. The female reproductive organs include a pair of ovaries. The ovaries are situated near the kidney. Important. Ovary is situated near the kidney. Testis also is situated near the kidney. But testis and kidney has a connection. Ovary and kidney don't have a connection because female frog is very independent. Very independent. Okay? Very independent. Cloaca is a common opening for your digestive system, reproductive system and your excretory system. Understood? Yes? Okay, a pair of ovaries which are present near the kidney but they have no functional connection to the kidney. And there is a pair of oviduct that arises from the ovaries and open into the cloaca separately. A mature female can lay up to 2,500 to 3,000 ova at a time. This is why Mr. Bean's entire apartment was filled with frog because he thought it was some beautiful fish and he took and went home. Fertilization is external. takes place in water. Development involves a larval stage which is called the tadpole, and the tadpole will undergo metamorphosis. That means changes you can see gradually. From the larva to the adult, you can see the changes. Tadpole undergoes metamorphosis to form the adult. Any doubts in this? Any doubts in this? Is this clear? Is this clear? Yes or no? Okay. Yeah, don't be Mr. Bean. Okay, 2000 to 2500 to 3000 over. 3000 over. Right? Yes. So, this is the structure. Look at the structure, students. Look at. I feel like I'll break this AC, students. It keeps increasing by itself. It's like I don't care about the remote. I don't care what temperature you want. I will increase. So, students, this is the oviduct. Okay, you can see the oviduct. Okay, you can see the ovary. Inside the ovary, you can see all the ova that is going to come out, which Mr. Bean will pick it up. Now, from the oviduct, the separate opening. Can you see separate opening is there? And you can see the ureter also is having a separate opening. Right? Same as in humans. Okay? Separate opening. So, cloaca, cloacal aperture and the urinary bladder. It's both the structures will look like this one butterfly structure. Okay? Both of it will look like a butterfly structure. Clear? Done, done, done. Perfect. Anything that I have to teach you again? Anything that I have to teach you again? If everything is clear, please give me a thumbs up. Please give me a thumbs up. So, students, those who joined late, let me tell you, animal tissues topic also is here. Okay, the notes are here, handwritten notes. So, please study from there. Okay, please study from the notes because I did a recent class on that. So, I do not want to repeat it again and again. Okay, because here time is very precious. We do not have time to play around. Right. So, here I have put the animal tissues notes as well. Okay, animal tissues notes as well, and then it continues with cockroach. Okay, Sir's notes also is here. So, everything I will share it together too on your Telegram, sorry, on Vedantu NEET English Telegram channel. If you want to join mine, this is Biology by Gopika. Thumbs up. Done, done, done. Okay. Congratulations. Today also most of you stayed. Yesterday most of you stayed for 8 hours. Today most of you stayed for almost 5.5 hours. So, good job on that. Okay, extremely good job on that. Use of cloaca is that cloaca is a common opening, a small chamber, a common opening for fecal matter to come out, urine to come out, as well as the sperm to come out. Understood? That means reproductive. Understood, Dhanush? Any doubts? Any doubts? Any topic? Any topic to be repeated? Anything like that? If everything is clear, can we end the session for the day? Can we end the session for the day? Okay, students, those who were asking, okay, those who were asking about the NEET crash course, this is the link. Okay, open the description box. This is the video you're watching. First of all, refresh and smash the like button. Okay, like the video, and as you move down, you will see that NEET 2025 crash. Click on the crash. Okay, you will see the prices. The prices are same itself. That is, oh my God, students, the price decreased. Okay, the price decreased today. Okay, 999 for 4 months. Okay, and crash plus QR code is only 2000 rupees. Okay, I am so excited. I don't know why. And 5000 rupees for crash flashcard all India test series plus doubt solving. Okay, batch is starting on 31st March. You have few more days. So before that, you join. And what are you going to get? You're going to get live classes, in-class doubt solving, quizzes will be there, leaderboard will be there, discussion of the practice question papers, and every week you will have the mock papers. Okay, then same thing will happen here, but here you will get the flashcard. This is the best one that you can buy because you will get a hard copy of the flashcard, and in your last minute, especially in the month of April, no, flashcards are going to come so handy to all of you that you can do all the revisions. So quickly take this because you will get, even if you don't want to watch the batch classes, you will still have repository of all the classes. Any classes you miss, you can always go back and check, and even if it is in a different language, you can check. Okay, so yes, 500 per month, you can understand. So it's starting on 31st, you will get proper one month 4 days ka training, full crash like revision. And here students, you have crash plus flashcard plus all India test series plus doubt solving, unlimited doubt solving. Okay, clear? So the link is right here. Link is right here. Okay, go just below. You'll see NEET 2025 crash DNA. This is for 2026. Okay, So that's it for today's class. Thank you so much for joining. See you all in next class. All the notes will be available on the Telegram channel. If you're new to the channel, do not forget to subscribe to the channel. And if you enjoyed the session till the end, quickly like that video and also comment down if you have completed the second day of our challenge. That is, today we have completed another, we have achieved another, 20 marks from these chapters. So thank you so much, students. Take care. Bye-bye.